All right, good morning. So we'll go ahead and, seeing that it's just a little bit after 8 o'clock right now, we'll go ahead and get started with the FDA Workshop this morning. I want to start by thanking everybody for coming out to the Focal Therapy Society meeting just a little bit early for this FDA Workshop. We have some folks from the FDA who have come downtown to give some presentations. We're hopeful that this will have some good discussion, good question and answer sessions, and good content being presented here. I'd like to first start off by welcoming Mark Antonino from the FDA, the Center for Devices and Radiological Health. We also have Daniel Suzman from the FDA Center for Drug Evaluation and Research, and also Michael Brown from the FDA Center for Devices and Radiological Health. They'll be giving their presentations this morning, and they'll also be part of our discussion panel for folks to ask questions, and hopefully we'll have good, stimulating conversation with the content that's being presented. So I'll start off and hand it over to Mark Antonino. He is in charge of the urological section for the FDA, and so he'll just start with some opening comments. Thank you.
Thank you, Dr. Rothberg. On behalf of the FDA, thank you for having us. Thank you for having the opportunity to participate in the workshop. Our mission at the FDA is to protect and promote public health. In being here today, we hope to help fulfill that mission. The presentations today hopefully will help provide considerations for evaluating clinical trial designs used to evaluate the risk and benefits of focal therapy treatments for prostate cancer. So we have a lot of speakers, and we hope to have a great conversation. Thank you again.
All right, so excellent. So we'll first ask Dr. Can anyone hear me okay? Does the volume need to go up a little bit? Good. All right, perfect. So we'll ask Dr. Michael Brown from the FDA to come up and give the first talk. He's going to give sort of an overview of the FDA, just in terms of terminology, sort of the alphabet soup of terms and stuff that are used for the FDA, as well as some additional content that he'll present on sort of device considerations with respect to the regulatory aspects of focal therapy devices. Thanks. Is everyone able to hear me okay? Okay. Oh, all right. Well, we'll wait for that to be figured out before we go ahead and get started, so that way everyone will be able to hear the content. Should I proceed? Let's wait for just a minute and allow for the AV folks to sort of optimize things.
All right. Thank you for the introduction, Dr. Rothberg. I'm Mike Brown, urologist and clinical reviewer for the urology devices team of the FDA's Center for Devices and Radiological Health. And I'm going to present a brief overview of the considerations for prostate cancer treatment devices today. Catch up with myself here a minute. Okay, so first I'll highlight the statutory basis of FDA device regulation. After that background, I'll go over the current direction of focal therapy devices and the challenges faced from a regulatory standpoint. Lastly, we will discuss what the future directions could be for prostate cancer studies, and end the presentation with questions and discussion, which I think will come after the second talk. With the information presented, I hope to stimulate a discussion on the prostate cancer clinical studies. Although the FDA was formed in 1906, until 1976, FDA had very limited authority over medical devices. For regulatory purposes, FDA treated devices as drugs in these early years, but did not have pre-market review authority. That changed in 1976 with passage of the Medical Device Amendments. Coming on the heels of injuries due to the Dalcon Shield intrauterine device, these amendments provided the FDA with the wide range of authorities, including the authority to regulate pre-market review of new devices. Devices on the market on or before 1976 were considered to be grandfathered and permitted to remain on the market. These regulatory activities are currently performed by the Centers for Devices and Radiological Health. Importantly, the statute provided for 3 risk-based classes of medical devices, each requiring a different level of regulatory scrutiny. A risk-benefit analysis is typically performed where both safety and effectiveness are evaluated. Class 1 devices are considered low risk. The FDA believes these risks can be mitigated by general controls, which are basic requirements that typically apply to all devices and include good manufacturing practices, labeling requirements, adverse event reporting, reports of corrections and removals, and establishment of registration and device listings. Class 1 devices are generally exempt from FDA pre-market review. Class 2 devices are moderate risk. The FDA believes that sufficient information is available such that the risk can be mitigated by both general and special controls. Special controls are device-specific requirements and include special handling, special labeling, testing, and adherence to industry and other standards. Special controls are written into the regulation of that device. FDA pre-market review of Class 2 devices is generally required. De novo, on rare occasions, are also classified as Class 1 devices, or can also be classified as Class 1 devices. De novo submissions may include bench testing, preclinical, and clinical testing. Class 3 devices are considered high-risk devices. These devices support or sustain human life, are of substantial importance in preventing impairment of human health, or present a potential unreasonable risk of illness or injury. The regulatory controls are needed to ensure reasonable assurance of safety and effectiveness. Insufficient information exists to mitigate the risks with general and special controls alone for Class 3 devices, although general controls continue to apply. Valid scientific evidence in the form of clinical data is typically required in the pre-market approval or PMA application. The most commonly used pre-market pathway is pre-market notification, more commonly referred to as 510K. The 510K is risk and class dependent. As previously stated, Class 1 devices are typically exempt from requiring 510K review. Class 2 devices are required for the 510K or de novo pathways. The 510K pathway is appropriate for so-called Me Too devices, where a predicate device with the same intended use and frequently the same technology has been identified. A predicate may have been previously cleared through the 510K or de novo process or grandfathered by the Medical Device Amendments of 1976. The regulatory standard is such that the FDA must find the new device substantially equivalent for the predicate to clear its pathway to market. Where there are differences in technology, those differences should not give rise to new questions of safety or effectiveness. The de novo pathway was introduced later as a means of approval for innovative but lower risk devices. Since it's an innovative device, no appropriate predicate can be identified. Without an appropriate predicate device, all new devices are automatically classified as Class 3 devices. The de novo pathway allows for reclassification of an innovative Class 3 device to Class 2 or even Class 1 if the FDA is assured that general and special controls can mitigate the risk appropriately for reclassification to a lower risk category. A de novo request is granted to market. Class 3 high-risk devices undergo the more lengthy and rigorous pre-market approval or PMA process, where new clinical data is almost always required. This slide provides a quick overview of the regulatory history of 2 existing devices indicated to treat prostate cancer. The first example is brachytherapy seed implants. Brachytherapy seed implants were on the market prior to 1976. These early devices were large radioactive pellets that were used to treat a wide range of solid tumors, including prostate cancer, and needed to be implanted in an open surgical approach. In 1976, this category of devices was placed into Class 2, meaning that subsequent versions will require 510(k) clearance. Over time, multiple incremental modifications were found to be substantially equivalent based on comparison of radiation parameters. These modifications involve reduction in seed size and the introduction of needle-loaded seeds, which together allowed implantation via a percutaneous approach under ultrasound guidance. These modifications were also aided by advances in dosimetry planning software. Like brachytherapy seed implants, early cryosurgical systems were on the market before 1976 as well. These early systems had large cryoprobes and were indicated to ablate soft tissue in a variety of medical specialties. Of note, a marketed pre-76 urological use of cryosurgery included treatment of prostate cancer, including transurethral placement of a single probe. After the medical device amendments, cryosurgical systems were classified into Class 2. Around 1990, smaller needle probes were developed, leading to the evolution of probe arrays that could be percutaneously placed within the prostate under ultrasound guidance. Additionally, several advances in cryogenic technology occurred during this time. These incremental changes were 510(k) cleared, primarily based on similarities in thermal and tissue ablation characteristics. The regulatory history of external beam radiation therapy, also frequently used to treat prostate cancer, is similar to that described here. It's important to note that other than radiotherapy and cryosurgery devices, no new prostate cancer device technologies have been approved in the U.S. since 1976. For a device with a prostate ablation tool claim, the main objective is to demonstrate the safety and effectiveness of the device for its intended use as a surgical tool for ablation of abnormal prostate tissue. We provided a link to the tool claim guidance here, as you can see. The devices with focal ablation tool claim are reviewed as 510(k)s. Special controls have been put in place to ensure reasonable assurance of safety and effectiveness. An example of this is clinical data. The images referenced here represent the standard of care currently available for focal therapy. Focal therapy treats localized prostate cancer. Note that to date, focal HIFU does not have a specific cancer indication, but only the tool claim. So how do the characteristics of focal therapy studies for tool claim compare to those for prostate cancer? The national, let's see, did I back up? Okay. The National Comprehensive Cancer Network, NCCN, defines intermediate risk prostate cancer as having are essential to demonstrate that the devices can effectively manage prostate cancer. For prostate cancer treatment studies, the sample size is expected to be statistically powered. It is also essential that there is long-term follow-up to assess freedom from salvage therapy, freedom from Gleason grade group 2 greater than or equal to 2 lesions, etc. Therefore, follow-up for several years seems appropriate. For example, 3 years minimum to assess a primary endpoint. It is ideal for the endpoints of the study to be specific to prostate cancer with the goal of complete ablation of the index lesion. The Prostate Imaging Reporting and Data System, PIRADS, particularly version 2.1, is the linchpin of decision-making of MP-MRI assessments of the index lesion to biopsy and or treat not only in the clinical world, but in the regulatory world as well. The PIRADS category refers to the likelihood of clinically significant prostate cancer, where 1 is equal to highly unlikely, the biopsy would be highly unlikely to show clinically significant prostate cancer. 2 would be unlikely, 3 indeterminate, 4 likely, and 5 highly likely. And this slide also is a tough angle to work from, but the slide also shows how the MP-MRI, the various modalities and parameters play into the PIRADS categorization. For example, here you can see that diffusion-weighted imaging 1 through 3 categories would usually play into a PIRADS 2 designation, whereas higher diffusion-weighted imaging readings would play into PIRADS 3. Similarly, dynamic contrast enhancement, when it's negative, would play into PIRADS 3 or less, and when it's positive, would be 4 or greater. The PIRADS version 2.1 regarding the peripheral and transitional zone lesions were subsequently updated. Changes were made to include ADC, which is the apparent diffusion coefficient, which measures the diffusion of water molecules. This was done to increase the sensitivity and specificity of the scale to reduce the number of indeterminate readings. In order to demonstrate clinical benefit for prostate cancer management, the clinical studies need to define adequate objectives, intended patient populations, establish meaningful effectiveness endpoints, and comparators. Some of the general considerations for these cancer management studies and endpoints that need further discussion are listed in this slide. The considerations listed are based on our current thinking and, again, are not legally binding. The objective of any management study is downgrading of higher grade prostate cancer, for example, Gleason grade group greater than or equal to 2 to Gleason grade group greater than or equal to 1, and returning patients to active surveillance. To adequately estimate the adverse event profile with clinically meaningful precision, including the incidence of infrequent device or procedure-related complications, we believe that the clinical studies need to be statistically powered. Ideally, a sample size of approximately 200 subjects or more seems appropriate. What is the ideal patient population for these clinical studies? What is the ideal lower and upper age limit? Similarly, what is the ideal prostate gland size to be considered for these clinical management studies? What are the considerations that go into determining whether a particular focal point therapy modality is appropriate for a patient with a prostate smaller than 20 cc's or larger than 80 cc's? Is it appropriate to include patients with PSA values less than or equal to 20 nanograms per mil in clinical management studies? Or should there even be a higher upper range? For these studies, do we only consider localized tumors with tumor node metastases, T&M score of T2B, negative nodes and negative metastases, lesions or less? The NCCN guidelines state that T2B and T2C tumors can also be treated. Some of these lesions could be amenable to focal therapy. Given that some of these lesions are very large, that is to say centimeters versus millimeters, does the Focal Therapy Society agree that T2B and T2C tumors can also be treated with focal therapy? Is it appropriate to include Gleason grade group 2 favorable patients only? What is the appropriate study duration for these studies? Follow-up is essential for these studies in order to assess freedom from failure. Therefore, it may be appropriate to have a 3 to 5 year study duration, 3-year primary endpoint, or with a 5-year follow-up. The endpoints appropriate for these studies are usually cancer-specific. For example, biochemical survival, progression-free survival, overall survival, or histologic progression from Gleason grade group 1 to Gleason grade group greater than or equal to 2. Or should we use surrogate endpoints, such as radical intervention-free survival, or freedom from systemic therapy, or freedom from salvage therapy, since these endpoints can be measured on a shorter time frame? As depicted in the image, there may be more than one index lesion. That is, more than one lesion of Gleason grade group greater than or equal to 2. When there's more than one index lesion, what do we do in the context of clinical studies? Do we stick to one index lesion paradigm, or do we allow treatment of more than one lesion? If we decide that more than one lesion is acceptable, then how many lesions do we consider acceptable? Another problem with having more than one index lesion is discussed in this slide. Is there a role for focal therapy techniques for whole gland curative therapy? Some of the cleared HIFU devices for tool claims have whole gland therapy capability. The potential advantage of focal therapy techniques is curative therapy for prostate cancer that does not involve the significant morbidities of classical radical therapies. Radical therapies being nerve-sparing radical retropubic prostatectomy with lymph node dissection or whole gland radiotherapy. However, focal therapy techniques also have some potential disadvantages, such as a possible sacrifice of some degree of effectiveness. The corollary here is there advantage to sub-whole gland therapy, which is whole gland therapy minus the capsule or some margin within the capsule. Or another way of looking at that, is there any clinical advantage in extirpating some of the Gleason grade group 1 lesion along with the index lesion, or effectively debulking the Gleason grade group 1 lesion? The practical aspect here is that there are devices under review that tout this capability. So what is the appropriate management of both Gleason grade group 2 unfavorable and Gleason grade group 3 intermediate risk prostate cancer grades? The problem from the standpoint of the FDA is the patient safety concern from exposing subjects who are usually referred for standard of care treatment, which is to say radical therapies, to be referred instead for focal non-curative prostate cancer management. Our concern with Gleason grade group 2 unfavorable and Gleason grade group 3 populations is that these groups are ordinarily referred to standard of care curative therapy. Therefore, is there a concern that if these patients are included in a focal therapy study design, that we may be unethically exposing them to undertreatment? Some possible solutions are discussed in this slide, such as using a comparative curative therapy, such as radical therapies, or a performance goal representative of curative therapy. Demonstrating through valid scientific evidence, for example, literature or real-world data, that Gleason grade group 2 unfavorable and or Gleason grade group 3 patients perform nearly as well over the long term without radical therapies as Gleason grade group 2 favorable patients. And of course, that could be a high bar. So future directions. Is there a future role for focal therapy in high or very high-risk patients with what otherwise appears to be localized disease, that is, Gleason grade group 4, Gleason grade group 5 disease? Does the expected decreased morbidity lend some justification for treating these patients? Questions to consider. The devices used for focal therapy are categorized by FDA based on the indications as either for management or curative intent. Does the Focal Therapy Society have any plans for developing any guidelines or definitions related to the different types of treatment goals? Our concern with Gleason grade group 2 unfavorable and Gleason grade group 3 populations is that these groups are ordinarily referred to standard of care curative therapy. Therefore, is there a concern that if these patients are included in a focal therapy study design, that we may be unethically exposing them to undertreatment? What are the considerations that go into determining whether or not a particular focal therapy modality is appropriate for a patient with a prostate smaller than 20 cc's or larger than 80 cc's? Okay. I've gotten myself lost here. See what we can do to get straightened out. Is there an ideal time frame for when biopsy should be done after patient enrollment, which we consider to be the time of consent, but before treatment? The NCCN guidelines state that T2B and T2C tumors can also be treated. Some of these lesions could be amenable to focal therapy, given that some of these lesions are very large, that is to say centimeters versus millimeters. Does the Focal Therapy Society agree that T2B and T2C tumors can also be treated with focal therapy. And I think after the next talk, we can have questions and discussion. Thank you.
All right, thank you very much, Dr. Brown. So I'd like to next introduce Dr. Daniel Suzman from the FDA, who is going to give some additional discussion. He is from Center for Drug Evaluation and Research, and so in addition to giving some regulatory considerations, also some statistical considerations for future focal therapy trial planning. Thank you.
All right. Good afternoon. My name is Daniel Suzman. I'm a medical oncologist and a deputy director within the Division of Oncology 1, which covers drugs involved in the treatment of genitourinary cancers, as well as breast and gynecologic cancers. Today I'm going to cover regulatory considerations from the drug and biologic side, which has some differences with the regulation of devices that we just heard about. So prostate cancer is a disease that has had a high degree of overtreatment. If the treatment of early prostate cancer was as simple as that of melanoma, where the early skin lesion is removed through local surgery with very little morbidity, there'd be very little need to focus our discussion on the low-risk local prostate cancer study population. However, curative treatments for localized prostate cancer can result in significant degrees of morbidity, including urinary and sexual dysfunction, which are life-altering and often irreversible. Active surveillance has increased in its use over the past few years, which is an impressive achievement given the high degree of anxiety men can have in the setting of known low-grade prostate cancer. However, a proportion of men on active surveillance do go on to have objective clinical or pathologic progression, which will trigger definitive radiation therapy or prostatectomy. The agency has seen interest in developing therapies, including focal therapies, biologics, and even drugs, in the active surveillance setting to delay or prevent pathologic progression and subsequent morbid surgery or radiation. Importantly, the benefit of this approach is the delay or prevention of the morbidity of radical prostatectomy or radiation therapy, and the delay in irreversible morbidity can be considered a clinical benefit from a regulatory standpoint. So in 2018, multiple stakeholders met at the FDA to discuss the development of therapies for localized prostate cancer, and I'm going to go over some of the topics, including endpoints and trial design that were discussed at that time. I'm also going to briefly discuss some of the challenges in the development of therapies for higher risk disease. So the outline of my talk is as follows. I'll briefly review the regulatory framework for approval of drugs and biologics and how combination products are regulated. I will discuss oncology efficacy endpoints and how they determine whether a product receives traditional or accelerated approval. We will then apply this to the challenge of trial design in the localized low-risk prostate cancer population. So there are different regulations for medical devices, and for my talk, I will focus on drug and biologic law. There are two laws that have led to the regulation of drugs and biologics. The Food, Drug, and Cosmetics Act for drugs require that they be safe and effective prior to marketing. The Public Health Service Act requires that biologic products be safe, pure, and potent. While these pieces of legislation have different language, the FDA Modernization Act minimized the difference in review and approval between drugs and biologics, and for all intents and purposes, safety and efficacy of drugs and biologics use a similar evidentiary framework. In our decision-making, we don't take into account issues related to cost. We also don't regulate the practice of medicine, only the products themselves. Just to touch briefly on the structure of the FDA Oncology Center of Excellence, this is an overarching center that fosters unified interaction between three FDA centers: the Center for Biologics Evaluation and Research, or CBER, the Center for Drug Evaluation and Research, or CDER, and CDRH, the Center for Devices and Radiological Health. So the Office of Oncologic Disease sits within CDER, but we work closely with CBER for gene therapies or cancer vaccines, for example, and with CDRH for diagnostics and for the regulation of drug-device combination products. There are over 100 oncologists, surgeons, radiation oncologists, and other physicians within the OCE. Some products are combinations of a drug and device, or a biologic in device, and when this occurs, a decision must be made regarding which center will be responsible for the primary review, and critically important, which regulations will be applied, device or drug biologic. The Office of Combination Products will determine the primary review center based on the component of the combination that provides the primary mode of action, where the indication is the treatment of cancer. This means which component primarily affects the tumor. So for example, a cytotoxic drug given systemically that is activated by a device locally, such as high-intensity focused ultrasound or a laser. In this case, the cytotoxic drug would be the primary mode of action to kill cancer cells, and CDER would conduct the primary review using drug regulations. So in the US, there are two approval pathways that can be used to approve a drug or biologic: the traditional approval pathway or the accelerated approval pathway. The appropriate pathway is related to the primary efficacy endpoint of the trial. So traditional approval requires substantial evidence that the product demonstrates a prolongation of life, a better life, or an established surrogate. Common endpoints for traditional approval include overall survival or large, clinically meaningful improvements in progression-free survival or disease-free survival. Accelerated approval is an expedited program which allows for earlier clinical endpoints other than irreversible morbidity or mortality to be used, which are reasonably likely to predict clinical benefit. The endpoint most used for accelerated approval in cancer trials is durable response rate in single-arm trials. Because there is residual uncertainty regarding meaningful clinical benefit, a post-marketing clinical trial is conducted to verify benefit, typically using a randomized trial with PFS or overall survival. So with that in mind, let's talk a bit about trial design, efficacy endpoints, and how I evaluate the strengths and limitations of an efficacy result. So generally, there are two types of trials we evaluate in OOD: superiority trials and non-inferiority trials. There are also equivalence trials, such as those for generic drugs, which I won't discuss today. The assumption for a superiority trial is that the product is superior to either a placebo or an active control. And for a non-inferiority trial, we're trying to determine that the product is not worse than an existing treatment. And I want to discuss that in the context of focal therapies, because there are highly effective standards of care for intermediate and higher risk localized prostate cancer, and it seems unlikely that focal therapies would be superior to these whole gland treatments with respect to oncologic outcomes such as overall survival or metastasis-free survival. And so focal therapy may need to demonstrate non-inferiority with respect to oncologic outcomes and superiority with respect to quality of life outcomes. There are several assumptions that go into a non-inferiority trial, including that the control effect is constant over time and populations, that the control actually has an effect, and that you can reliably estimate that effect. So with that said, non-inferiority trials may be very challenging to implement for focal therapies. Treatments have changed over time, particularly for radiation therapy, where higher doses and improved coverage has impacted oncologic outcomes, particularly for higher risk prostate cancer. And as imprecision in non-inferiority trials biases towards a finding of non-inferiority, use of endpoints that may be subject to imprecision, such as those dependent on relatively infrequent imaging, are problematic. So a non-inferiority trial in this setting is likely to be very large, and there are challenges interpreting time-to-event endpoints other than overall survival. So with that in mind, I'll switch the focus back to lower risk disease, considerations for different efficacy endpoints, and how I evaluate the strengths and limitations of an efficacy result. The strength of an efficacy result can be evaluated by looking at what is measured, how it is measured, and how much of an effect is demonstrated. What is measured is important for the reasons I discussed regarding direct measures of clinical benefit, like prolongation of life or improvements in symptoms or function, since they are considered more clinically meaningful than surrogate endpoints. However, one must also look at the measurement characteristics of the endpoint. How accurate and how much variability does the measure have? How susceptible is it to bias? And how accurate is the timing of the event? Some endpoints, like patient-reported outcomes, are strong in what they are measuring, direct benefit, but may have challenges in their susceptibility to bias in open-label settings. Finally, how much of an effect is important? Large effect sizes can mitigate some of the weaknesses of an endpoint. For example, an 80% response rate with a median duration of 18 months in a third-line pancreatic cancer single-arm trial with acceptable safety will be challenging to run a randomized trial to confirm an OS benefit. On the other hand, even overall survival can have a statistically significant result of a 1 week, which is not likely to be considered meaningful in the setting of burdensome toxicity. So looking at this more closely, I provide a few examples of increasing levels of interpretation and subjectivity in endpoints that can add risk for bias and variability. In this figure, the lowest interpretation would be survival. One is either dead or alive. Progression-free survival for measurable tumors like breast and melanoma requires a little more interpretation and can be subject to investigator bias. Radiographic PFS in prostate cancer requires even more interpretation using bone scans. Finally, preventing a morbid procedure requires a decision to act by the investigator and patient. For instance, palliative radiation is one component of the composite endpoint skeletal-related events. But for this event to occur, the patient must report pain to the healthcare provider. The healthcare provider must assess the pain as focal and related to a known bone lesion that is amenable to radiation. The patient and doctor then need to agree to radiate the lesion. This is particularly relevant to trials in the local cancer setting, where the primary treatment is morbid, such as bladder and prostate cancer. Preventing the morbidity of the procedure is a real clinical benefit, but the endpoint has such a large amount of subjectivity and has risk for bias that increases regulatory uncertainty. Another component of the measurement characteristics of an endpoint is the timing of the event. For time-to-event endpoints like PFS and OS, timing of the event affects the magnitude of the result, which is used to assess risk and benefit. OS is known to the day, but PFS is only as accurate as the frequency of the scanning intervals. So looking at the strengths and limitations of overall survival is clearly a strong endpoint. It's a direct measure of clinical benefit, which is a prolongation of life. It has low risk of bias, as the event requires no interpretation, and the event, death, is known to the day. It also captures both efficacy and safety. The challenge, as we have been discussing, is that this is the last event in the natural history of the disease, and this affects the feasibility of the trial. So let's finish this talk by applying the regulatory framework to the setting of low-risk localized prostate cancer. Active surveillance has been very successful in reducing overtreatment for low-risk prostate cancer, but as I mentioned, patients do go on to receive morbid procedures. There are several challenges for trials in active surveillance populations. First, endpoints that have led to FDA approval in prostate cancer, overall survival, and metastasis-free survival, are impractical. Second, delaying a morbid procedure like curative radiation or radical prostatectomy is clinical benefit, but as I mentioned, it's subject to bias and increases regulatory uncertainty. Third, PSA anxiety can drive patients to go on to receive definitive whole gland therapy without pathologic local progression. Even if the intervention delays prostatectomy or radiation, it is delaying the morbidity, not the procedure itself, that is clinical benefit. And so acute and subacute toxicity of any intervention in the active surveillance space must be well tolerated. Lastly, there is concern that delay of a needed prostatectomy or radiation therapy treatment could reduce cure rate and have higher morbidity if the curative therapy is given after an earlier focal therapy. To illustrate the issue, I have included some of the clinical outcome and biomarker imaging endpoints on a timeline I've adapted from the Hopkins Pound series of patients post-radical prostatectomy. Importantly, these patients are higher risk than an active surveillance population, and so these timelines will be even longer. As you can see, clinical outcomes in blue that are not preventing or delaying morbid procedures, namely metastatic pain, skeletal-related events, and overall survival, occur over a decade from PSA relapse, even in this higher risk population. Metastasis-free survival has been used to approve a new molecular entity. However, this endpoint is also very likely to be hard to power and will take many years to mature. So looking at the strengths and limitations of various clinical outcomes from this timeline, overall survival is a direct benefit and has low risk for bias, but is impractical. Metastasis-free survival is an important endpoint that defines an important change in clinical state and risk for cancer morbidity or death, and has recently been used to support the traditional approval of a new molecular entity, apalutamide, for patients with non-metastatic CRPC. MFS is a strong endpoint. However, as we have shown, this endpoint also occurs late in the natural history of the disease and would also require a very long and large trial. We also don't yet understand how the clinical meaningfulness and feasibility of capturing this endpoint will change in the era of PSMA PET scans. delaying or preventing definitive local therapy and its associated morbidity is considered clinical benefit. However, this endpoint is challenging due to the risk for bias, which is compounded by patients who often have high levels of PSA anxiety. On the other hand, this endpoint is much more feasible than OS or MFS in this setting. Patients are monitored on active surveillance, and a portion will have biopsy or other clinical progression that triggers a recommendation to undergo curative treatment. Objective local progression has lower risk for bias and is feasible. However, objective local progression is not a measure of clinical benefit in and of itself, and would therefore be considered a surrogate endpoint, which would be more appropriate for accelerated rather than regular approval. So regardless of all these challenges, we are receiving trial designs in this space and have heard from some investigators and patients that the active surveillance population is an area where therapies could delay or prevent morbid procedures and should be studied. The following few slides present a straw man trial design for consideration that hope to address the issues and stimulate discussion. With respect to efficacy, OS and MFS are impractical, and delaying morbidity from local progression is meaningful but subject to bias. One path forward could be to use a pre-specified definition for local progression based on existing active surveillance protocols as the primary endpoint with supportive data on clinical benefit provided by delaying or preventing morbid curative local therapy. So in this case, both endpoints are feasible. The clinical benefit is demonstrated by delaying the morbid procedure, and the bias is mitigated by using the objective pre-specified trigger to come off active surveillance via biopsy results and other objective signs of local progression. So I should pause here and note that, again, it is not the delay or prevention of the curative local therapy itself that is the benefit. It is the delay in the morbidity associated with that treatment. If the curative treatment was an outpatient skin surgery, as is the case for early-stage melanoma, this design would not work because the curative therapy has very little morbidity. Because of this, the key morbidity of curative local therapy, urinary and sexual dysfunction, must be measured well. PRO tools exist for both of these morbidities, and the adequacy of the PRO tools for regulatory purposes should be discussed with the FDA. The data from PRO can be descriptively analyzed for safety, but could also be another statistically tested key secondary endpoint with alpha allocation to confirm the findings from delay in the procedure itself. As an additional consideration, definitive therapies themselves continue to improve and may have less morbidity than in the past. In addition to the efficacy concerns, for trial to support a favorable risk-benefit, the acute and subacute safety of the intervention has to be acceptable. The primary clinical benefit of a trial in active surveillance would be to delay or prevent morbidity. If there's a significant morbidity for the local intervention itself, then any benefit in delaying progression to curative whole gland therapy would be abrogated. Acute and subacute safety and tolerability would have to be carefully measured. In addition to standard CTC-AE safety surveillance, sexual and urinary morbidity must be carefully measured using well-defined and reliable patient-reported outcome tools. Other patient experience data, including use of a symptom library, like the NCI's PRO CTC-AE, coupled with an overall side effect summary measure, could also add important complementary information to the safety assessment. Demonstrating a statistically significant delay in objective progression coupled with delay or prevention of definitive whole gland therapy and acceptable acute and subacute safety and tolerability using clinical and patient-reported data could be one path to traditional approval. However, important residual safety concerns would remain. The potential for reduced cure rates and synergistic post-procedural morbidity for those who go on to definitive whole gland therapy would require further study. Post-marketing safety studies can assess long-term safety outcomes, including biochemical occurrence, use of salvage treatment, descriptive data on metastasis-free survival and overall survival to ensure no large decrement, and patient-reported outcome measures of urinary and sexual dysfunction. Patient-reported quality of life could also be supportive. So to summarize, this schema demonstrates one potential trial design that could be done for a focal combination therapy with a drug or biologic primary mode of action. Patients eligible for active surveillance would be randomized to focal therapy plus active surveillance versus active surveillance alone. The primary endpoint would be objective local progression, and the key secondary endpoint would be time to local curative therapy. Acute and subacute safety would be carefully assessed, including patient-reported outcomes to assess tolerability, urinary dysfunction, and sexual dysfunction. To assess long-term safety, a post-marketing requirement would be long-term follow-up to assess for biochemical recurrence, use of salvage therapy, and continued monitoring of urinary and sexual dysfunction, as well as serious adverse events. So there are several design and conduct considerations here. Objective criteria for local progression would need to be defined. Assessments of biopsy, MRI, PSA monitoring, and pathology review would need to be symmetric. Patients would need to be educated regarding PSA anxiety to minimize going on to curative treatment in the absence of pathologic local progression. So the definition of local progression can be determined by reviewing standard practice and active surveillance programs. However, there is some heterogeneity in the different programs and newer technology that is beginning to be used, such as MRI. These criteria would need to be carefully considered. So I'd like to conclude by saying I can see both sides of the argument for whether there or not there is an unmet need for patients with low-risk localized prostate cancer undergoing active surveillance. On one hand, you can argue patients should continue to do active surveillance, and we should do a better job at preventing local treatment due to PSA anxiety, and no therapies need to be developed. On the other hand, we know that there are a subgroup of patients who do progress and require definitive therapy. If a focal therapy could demonstrate delay in local progression, delay in morbid curative local therapy, and have a favorable safety profile demonstrating lower rates of urinary and sexual dysfunction, and provide data that long-term cure rates are not adversely affected for those who do need curative therapy, then we might save even more patients from overtreatment associated with irreversible urinary and sexual morbidity. If one agrees that this space is an unmet medical need, does the trial design framework adequately address the efficacy considerations, and what safety and other endpoints should be monitored in the post-marketing requirement for long-term follow-up? Lastly, there are further challenges with use of focal therapies in higher-risk patients where they will likely need to be compared to whole gland therapies. Thank you for your attention. I look forward to the discussion.
So thank you so much. We'll invite Dr. Brown to come back up. I'll open to questions and discussions. We had some excellent content that was presented by our colleagues at the FDA this morning, and so we'll have them come up for a question and discussion panel, and I'll go ahead and turn it over to anyone who has questions.
Hi there, Samir Taneja from New York. Thanks for those great presentations. They summarize a lot of the issues that we've dealt with. My question is really about risk stratification, because the FDA has been very reliant on the NCCN risk categories, which I think some of us would argue are a rather blunt tool. There's certainly risk overlap between unfavorable and favorable, and the current definition of favorable probably forces us into designing trials, as you said, for people who maybe don't need treatment at all, at least initially. So the question is, why is there reluctance to use other nomograms, predictive nomograms, which are in the space that have been well-developed, well-validated, particularly those that use imaging, such as the Briganti nomogram, et cetera? And where does PSMA PET fit in now? Does that lower the bar if we can take an unfavorable intermediate risk patient and do better baseline staging? Does the FDA view that as perhaps more appropriate for a focal treatment or a trial design?
Well, I think that your point is well taken, and I don't think that there's any reluctance, particularly on the part of, at least in my experience, there hasn't been any reluctance on the part of the FDA to look at nomograms. As you pointed out, there are so many variables that can be considered in this particular issue, and I think it's fair to say that it's an issue of active discussion, and that no real consensus has been decided upon at this point, which is part of the reason for this presentation, to hopefully stimulate the kind of discussion that you brought up. For me personally, I certainly would not have a problem looking at those sort of factors. And to your point, I think that I think it's a good point, and that may be something I'll look into going forward.
No, please, you first.
You were first.
Thank you. Michael Gorin from the Icahn School of Medicine in New York. So question specifically for Dr. Suzman. So I really liked hearing you talk about the active surveillance, low-risk prostate cancer space and questioning whether or not there was a role for potentially focal or some form of therapy that delays progression to radical treatment in those patients. However, many of us were present in the room in 2020 at the ODAC meeting where specifically that question was reviewed with respect to Stevia's new drug application for TUCAD. And the meeting, I think, was like 14-2 against using a therapeutic in that space for those patients. Given what you said today, how do we reel that back in when we have an ODAC meeting on the record that specifically said we can't have a therapy in that space?
Well, I mean, I guess I would challenge the notion that you can't. See, I'm not sure this microphone is going on. You know, that space is necessarily off limits. I think there were FDA had concerns around the TUCAD design, you know, specifically around the adequate measurement, adequate follow-up subsequent to local therapy, you know, for urinary sexual dysfunction and so forth. You know, you can have discussion, you know, with the FDA around what the appropriate patient population, you know, is that may be appropriate for, you know, for therapy, whether that's, you know, a somewhat higher risk patient population than your, you know, 3+3 low risk or very low risk population.
Sorry, I was going to say, because this all seems very opaque to me, because from my view of those data and many of the urologists who were in those rooms, they accomplished exactly what you had said would be proposed, and that is showing a delay to radical therapy without harms with respect to urinary function and erectile dysfunction, but yet it was completely shut down. So, I mean, I'm very encouraged to hear you say that there's a potential in that space. It just seems to me like we're getting two different, you know, answers.
Yeah, I mean, I think you can go back and look at the FDA presentations from that, you know, from that time, and you know, and again, you know, an oncologic advisory committee is sort of advisory, and I think, you know, does not necessarily open or close doors fully. I think my recollection is there were concerns around, you know, the trial design, you know, having adequate follow-up data. As I mentioned, you know, the part of the issue there is making sure that the local therapy is substantially less toxic than whole gland therapy, and that you are actually preventing morbidity, you know, from subsequent therapies. I think, you know, there were concerns, and I remember Dr. Walsh commenting on the, you know, the very low risk population that he thought did not need to be treated at all. So I think there's still some debate around what the, you know, appropriate patient population is, and ensuring that there's adequate follow-up to ensure that you are actually demonstrating clinical benefit there.
It was very clever to have PCW in the anchor seat to speak first at the meeting. Thank you very much for responding.
Rafael Sanchez-Salas here. Just to add on what Dr. Garins mentioned, you have the TUCA trial, you have a real world data out there telling us not to treat Gleason 3+3, and then you have trials that are coming out of in Europe in which even 4+3 or oligometastatic disease is treated with focal therapy. So how are you planning to tailor your situation and go beyond this Gleason 3+3? It's just not perhaps the Gleason. We need to add up some selection imaging to be included in the trial and some selection to be included in the follow-up. So it's perhaps not just the Gleason that will make the difference in the future. So what were your expectations in terms of trial design? Because this slide that you presented is a great slide. It gives a message, but it gives a message that perhaps already was answered. And we have trials that are moving forward in a different direction, perhaps more faster than what we have been done in North America. So what are the expectations to tailor on that?
Well, the expectation is that we will move forward at some point in the hopefully near future with regard to these patients with more grim prognostic indicators. To some extent, we are limited by the data that the sponsors bring to us, and we certainly consider real-world data to the extent that it's clinically reliable and robust, sufficiently robust. Real-world data is certainly considered to be valid scientific evidence. So at the point that we see that these patients can be treated safely with focal therapy, we're not opposed to doing that. In fact, our whole goal is to help the clinical world move forward. But again, our mission is to protect the public safety, and so we have to have that reliable data available. As was mentioned earlier, that again speaks to the nomograms that include imaging and other parameters, and that may be something that we look into to help us facilitate getting some of these higher grade patients treated with focal therapy.
Simultaneously, we try to be cautious, and then in the private setting, they're not cautious. They're not necessarily cautious. The abuse of focal therapy that hurts the approach, and this definitely is creating a circle of extended discussion in which we have our peers from the radiotherapy side or surgery side accusing us of abusing of an approach that is not necessarily accepted, and it's not really what happens in the academic centers. So at some point, the middle ground needs to be defined so we can move forward in terms of how the therapy will be eventually make it to the gold standard.
Yeah, I mean, I agree with you completely, and to the extent that we can arrive at some consensus on which nomograms and factors, et cetera, that we're looking at, that kind of agreement, that kind of consensus is what helps us at the FDA move forward as well. But I agree with you completely. We're not here to stifle progress, but again, as I stated earlier, we're on the conservative side by the very nature of what we do. And so public safety is very high up on our list of priorities, but we're certainly not opposed to the ideas that you've posed. We want to assist you in moving forward, and that includes the higher grade patients.
Thank you.
Very briefly, I'm Sri Verganti from Chicago at Rush Medical Center. You laid out considerations, particularly in trial design, oncological and whatnot. I was a little curious about size as being a major factor, and we're all aware that size has impact on quality of life outcomes, which are certainly going to be impactful to clinical trial design. But I was interested specifically in the terms of trial design, how size exclusions at the extremes or whatnot could be built in. I do recognize that cautions at large extremes are important, but I would say the standard of care options are fraught with these same problems. So if you take someone at extreme sizes, they're going to suffer challenges from standard of care, and often are patients who were very interested in alternatives to the standard array. So if you could elaborate on that specifically, because the others seemed fairly straightforward in terms of the outcomes that we're measuring.
So when you say size, you're referring to the size of the prostate?
Yeah, exactly. So you mentioned, I think there were some size ranges like 20 to 80 grams.
Mm-hmm.
And, outside of that range, we should have particular concerns and things like that. And of course, with all patients with size extremes, this has impact, particularly on quality of life and impact of treatment. I imagine there'd be corollaries to comorbidities and whatnot. But I was curious if you could elaborate on that specifically as we come up with, inclusion criteria for our trials.
Yeah, well, thank you for that question, because, that's one of the objectives of this forum, is to generate some discussion around those types of issues. Certainly one of the things that I have considered is that, a man with a 300-gram prostate who has prostate cancer is just as entitled to treatment as a man with a 50-gram prostate with prostate cancer. The question becomes, what size range do we recommend? And I think that those issues that you brought up would be the types of issues that have to be considered. The relative morbidity of doing larger glands in the context of a clinical study. We understand that practitioners have the discretion to do whatever they feel is appropriate. We don't regulate medical practice, but within the context of placing patients at risk, frankly, in the context of a clinical study, we have to have some parameters, and the discussion becomes, you know, what is the appropriate parameter? To your point, we had said that, and this is based on what we've seen, basically. Basically, we've seen 20 to 80 cc prostate glands, but is 120 cc prostate gland more appropriate? Is a 200 cc prostate gland more appropriate? So, and we're open to those sorts of discussions. So I appreciate that feedback, and that is, again, something that we will take into consideration.
I'm Kerry Robertson from Duke Medical Center. This is a question for Dr. Suzman in reference to drug development versus device development. In usual parameters, we'll see drug development in the greatest degree of degrees of disease. So for a biologic or a chemotherapeutic agent, it'll go first to a phase 1, then a 2, usually in metastatic disease, maybe a more progressive last stage disease, and you look for effect, and then you dial it back to lower volumes, then finally to adjuvant therapies. So in a device development, we're asking the opposites, reverse logic. We're going for an early stage disease. Have you thought about using low volume, high grade disease as the initial candidate for device comparison in a randomized format with traditional therapy?
So, you know, I'll note that from a drug biologic standpoint, I think we have somewhat different regulations than on the device side, and different trial designs, different endpoints, you know, standards of evidence and so forth. So I think we're, you know, I agree that for metastatic disease, that's generally been the paradigm, although we have within OCE, something called Project Frontrunner, which is an interest in developing, you know, moving drugs a little further upstream earlier. But we do take safety considerations very strongly into account when we're dealing with a potentially curative disease that has an appropriate alternative therapy. So, you know, we don't think necessarily that the adjuvant setting is an appropriate place to do dose optimization. We'd rather see that done in a metastatic setting. You know, I think the questions around, you know, you know, the metastatic setting, the local setting are sort of somewhat different. I think we're open to if it, you know, if it's a, again, I think the concern would be if this is a patient who would likely require require curative therapy. I think we have higher safety considerations and demonstration of initial evidence.
I'm going to be devil's advocate because in high-grade disease and prostate cancer, our usual therapies are not always curative. We have a high failure rate. And
sure,
we don't admit that very often, but it's true. And when you're looking at technology which can be additive and potentially beneficial and with least morbidity, there's a possibility you actually have clinical benefit with an earlier achievement of OSS survival endpoints.
Yeah,
so just to raise the question, because we're stumbling over active surveillance in the long OSS periods, when actually you can go with low volume, high grade disease and a very well-defined small lesion. And we see this all the time. And there may be one core of Gleason 4-4 in a small prostate. And we wonder, how do we really should manage that, right? So, you know, with imaging, with improvement of imaging, we're probably going to be able to define very small high-grade lesions very accurately in the future, if not currently. So that would raise that point, and I think that's actually a reasonable trial subject population.
Sure. I mean, I think we're open to that with the appropriate evidence rationale. You know, that's a safe place to do. You know, and that you're not denying patients the chance of a you know, curative therapy. You know, again, a concern would be, you know, if you're giving this and it somehow precludes then, you know, the chance of cure, that would be a major concern. But otherwise, you know, if there's appropriate safety and, you know, at least some early efficacy data that you know, that this is an appropriate setting, appropriate device, I think we'd be open to that consideration.
Caroline Moore, University College London. Just to say, I completely agree with your idea of low volume, high risk disease. I'm really fascinated in the differences between regulation, particularly in the UK and in the US. And I was just wondering, in terms of real world data, is there a requirement for that real world data for your review to be in the US, or is international data also acceptable?
Sure. I mean, I can address it at least from the CEDAR perspective. I think the main consideration around real world data is that it be fit for use. I don't think that we have, and I think it would need to apply to the U.S. population, even if it came from other countries. And I guess I'm not sure. It sort of depends on what you're anticipating using this real world data for. But the fact that it comes from another country is not necessarily a problem as long as it would apply to the U.S. population.
Thank you. Yeah, it was really the registry data in the UK and other countries.
And
Go ahead, Mike. Go for it.
Yeah, I was just going to agree with Dr. Suzman that we certainly consider real-world data. And like U.S. data, the quality of the data, of course, is what's important to us in terms of the number of cases, et cetera, that are presented to us, the qualifications of practitioners, et cetera, which is generally not a problem with the European practice. But there are some things that we do need to see. For example, when we're dealing with out of the United States data, we do need to know that that data can be applicable to the U.S. population. That is, that's generally not a high bar, again, for most of the European data that we receive, but that is important. For example, some of the European countries don't have much in the way of an African-American population. Well, it wouldn't be African-American, it'd be black population. Which can be important, particularly when you're discussing prostate cancer, for example. So those are the types of things we look at. But no, again, real-world data generically is valid scientific evidence to the extent that it's clinically reliable and robust. We welcome that and certainly consider that, very strongly.
Thank you.
And sure, we have published a guidance document, which is our current way of thinking on the use of real-world data. As Mike said, it can be applicable. But just as as it's taking a step back, from the medical device side, again, there are no approved or cleared devices for focal therapy for prostate cancer treatment or management. So this is the opening of a discussion as these investigational device exemption trials get off to a start. This is a discussion for us to understand, how we can have a reasonable understanding that these devices, the risk, the benefits will be outweighed by the risks, and we can take that information into further account for marketing applications at the time. But to underscore again, this is the beginning of a discussion. So thank you for all the comments.
So I have a follow-up question. So we had some great sort of discussion and content presented on sort of comparing clinical endpoints and what would be most meaningful and most useful in consideration for these ablation devices as a tool versus for cancer management, but also with respect to different patient populations. So there was a good trial schema that was presented for, sort of maybe like an intermediate unfavorable risk patient versus someone who would undergo radical whole gland therapy. So with those considerations and knowing that we need more clinically meaningful sort of cancer-specific endpoints, do you guys feel as though, the endpoints that were discussed are applicable for sort of both of those target populations, or is there something that might be more specific or more appropriate for one target population versus another?
I mean, for a population where the standard of care is definitive therapy, and there's a high risk for development of metastatic disease, I think we'd want to see real oncologic endpoints. I think there may be this window in the active surveillance population, on the sort of the higher end risk, for focal therapy or sort of add on to active surveillance. So I think there are sort of two different paradigms, and in part it's some of what the community feels is the appropriate standard of care therapy as well.
Yes, and I would also add, certainly in the world of urological devices, it's obviously, as Dr. Suzman pointed out in his presentation, oncological endpoints are certainly the preferred endpoints. Unfortunately, we have to walk a line between what's preferred and ideal and what's practical. It's a difficult ask to have sponsors, for devices at least, come in and tell them, okay, well, we need a 10-year study in order to establish whether your device is safe and effective for this particular purpose. So, you know, hence comes the discussion of surrogate endpoints, which are not as clinically meaningful but are more practical. So it's a tightrope walk. I wish we had an answer for that, but it's something that we have to consider on a case-by-case basis, frankly, and anybody.
Okay, thank you. So we do have just a couple modifications to our schedule this morning. We'll break for a coffee break in just a second before we get into some of the more clinical talks for the second half of the program. Before I do, I just want to invite Dr. Peter Pinto to come up and just give some remarks. He is an investigator in the urologic oncology branch at the National Cancer Institute, and so he'll give some comments on this space as well.
Well, listen, there's nothing more to speak about. I mean, this was a wonderful session. I'm just here as the cheerleader, you know, honored to be the kind of the NCI liaison for the Focal Therapy Society, and to be able to work with Michael, who's, you know, the tears coming out of my eye as we lose him as a fellow, but he's moving on to greener pastures at Duke to start a career in this space. And to have our colleagues from the FDA come over and give really salient features about what we have to work on is very important. It just reminds us as a society that we have to work together with our colleagues, obviously, from the FDA and from industry to kind of push the field forward, because I heard a couple of times in the session this morning, you know, it's a little bit of a wild west out there. Without regulation, without codes, without guidance, any physician can take any device and do anything to any patient if there was a ablation approval for tissue. And we've all seen, as I can show that, you know, wonderful colleagues from all over the world coming here. Caroline Moore always giving thoughtful comments about this from her work in London with her team. And I just wanted to just remind us that. So going forward through the society, continued support, continued membership, opportunities to grow this will only help our patients. For my FDA colleagues, you know, a lot of pre-planning going into this. I know it's not easy coming down. As a federal employee of the U.S. government, travel is hard, even to D.C. when you work in D.C. You know, who's going to approve the Metro card? Who paid for that Metro card? I've been there, guys. I know what it's like. Our budget got shut down early this year, as you may have heard in the news, so I had to pay for my own travel today. I'm actually on annual leave to be here. Inside joke for my colleagues in the government. So I just want to say before we go to the break, thank you very much to our colleagues from the FDA for just taking their time to be here.
Let's do 9:40.
Okay, and then we'll resume here about 9:40 for our clinical section. Dr. Tom Palasic will head off that. Thank you.
Thank you.
All right, thank you, everyone. So we'll go ahead and we'll get started with the second half of the workshop this morning. We'll ask everyone who's finishing up their coffee break to come back in, take their seats. We'll give a few minutes for that. All right, so to complement some of the content that we had this morning presented by our colleagues at the FDA, we have a couple of folks to give talks on sort of the clinical aspects and considerations for focal therapy in this space. We'd like to start off, it's an honor to have Dr. Plasek, the president of the Focal Therapy Society, to come and give a talk and give an update on the Focal Therapy Society registry to start off this second half of the FDA workshop this morning. So thank you, Dr. Plasek.
I'd like to start by thanking everyone for coming and participating in this meeting. From my point of view, which is potentially biased, I'll give my disclosure, it's a little biased, but I think we need to do this for men's health going forward, and we like to form partnerships with industry and the FDA and see what we need to do to solve some of these issues to move the field forward in a very reliable fashion, I should add. Okay, so welcome everyone, and I've been tasked to speak on the registry. I have to give credit to the Spared group who started this. We had meetings with the FDA probably around 2017, 2018. And at that time, it was mentioned that we should have some kind of a device registry with real world outcomes, as was mentioned by Dr. Moore this morning. This would involve oncological outcomes, functional outcomes, quality of life, et cetera. And initially, there was some discussion of how best to do that. And, we took it to heart. That's my message for this morning. We took it to heart. We felt that the best way to do this would be to house the registry in a society, and this way no one would basically own it, and we can all use it and access it. So since that time, we've had the pandemic, and Spared really didn't take off as anticipated. I think there were very few in that registry, but the good news is that we now have formed the Focal Therapy Society in 2019, and we have our database up and running. So this is a registry. It's approved and certified by the Duke IRB. It is now officially open for enrollment. It is an open registry, meaning that you can use any focal therapy. You can put drug, device, treatment scheme. It could be image-targeted treatment only. It could be hemiablation, whatever you want. You just put this in the registry, and we'll document functional oncological outcomes, and also we've built in an MRI component for imaging. So why a registry? Well, there's many reasons. One is that we have to get some real-world data. I would mention that we recently wrote a paper coming from the Focal Therapy Society on the real-world evidence of whole gland, HIFU, and cryo over the last 20 years because it hadn't been done, and we felt that it was important to get real-world evidence and data out there, at least from a starting point. I mean, we have to have something to build on. So I really believe that clinical registries play a critical role for evidence-based medicine in addition to randomized controlled trials. I think you need both. This will provide real-world data on patient outcomes, treatments, interventions. It'll offer insights, perhaps insights that you don't get from RCTs, It'll look at safety concerns, efficacy items, and will hopefully aid in clinical decision-making. Now, registries are only as good as the data that people put in, so I would encourage everyone to participate. We need your help. So what are the objectives for the Focal Therapy Society Registry? Well, if you look at the guidelines, here's three governing bodies. The AUA says that ablation may be considered for appropriate patient care, but should be done in the context of a clinical trial or a registry. The EAU says basically the same thing. It should all be done in a registry. And NICE, essentially the same. They want registry data or randomized trial. Now, not everything that we do is a randomized trial, and what's interesting to me is even though you have a well-done randomized trial, we talked about one of them this morning, the TUCAT one, level 1 data, people sometimes don't use the data. So it's not that one's better or worse than another, but you just need all forms of outcome data. So these three governing bodies have told us that focal should be done in a registry. So now we have a solution. So the objectives, we want to capture high-quality data to determine oncological and genitourinary functional outcomes in patients undergoing prostate cancer ablation. There's a great need to collect this data, as mentioned. We want to also document that we're reducing morbidity compared to whole gland traditional options. And I think that this will aid in the addition of ablation therapies to guidelines, particularly where RCTs or their outcomes may be questionable. So with that, I would encourage everyone to support the registry. And I'll go over some of the numbers that you have right now, but I can tell you that Dr. Robertson, who's my partner and does predominantly HIFU, and I do predominantly cryo, we've done some other device trials as well, all the data is in the registry. So when someone says, well, you shouldn't be offering this outside of a registry, my response is, it's in a registry. So I would invite you to also put your data in a registry. If you want to participate, there's just a few minimal requirements. Number one, join and belong to the Focal Therapy Society. We are partnered with the Endourological Society, so essentially one fee gets you into both groups. So we have a very strong infrastructure. I think there's about 2,000 members with the Endo Society, and we have about 250 to 300 in the Focal Therapy Society. You need local IRB approval. If you're in private practice and you don't have that, Duke will support you with IRB approval, and then you have to sign a data user agreement to allow data to be shared. So not much to do to get into it. How do we collect the required data? The data is abstracted from electronic medical records. It includes diagnosis, treatment, surgery, follow-up every 6 months for 2 years, and then annually through 10 years, or as long as you want to go. We're hoping to perpetuate this. Collaborating sites may provide as much or little data as you're willing to. So if you are a detailed person and have the infrastructure to collect detailed information, you can put it in there. If maybe you don't, you have just general outcome, PSA, biopsy, you're welcome to put that in as well. And then we will follow these patients, or you'll follow the patients as part of the registry until death. Data to be collected, demographics, and then baseline clinical GU functional data, pre-op and follow-up imaging, biopsy information, details about whatever kind of ablation you're performing in terms of treatment, pattern, device, maybe if the patient had adjuvant, and secondary treatments. I know that we're getting into the realm that we're starting to use drugs as well. Like an example would be radiation plus ADT. So there's more of us now venturing into how do you make ablation more effective. So you may be using some kind of adjuvant or ablation enhancing agent. But this is not a biospecimen repository, so we won't be able to do that for you. Current status. In the U.S., you see we have 6 that are currently entering data: Duke, Northwell, MD Anderson, University of Michigan, Cincinnati, Chesapeake Urology, which is a LUGPA site. We have Singapore General Health. We have a site in Buenos Aires. And we have sites in the process of onboarding: Henry Ford, University of Colorado, Halo DX, Cornell, St. Peter's, University of Chicago, Mount Sinai Health, and in Canada we have University Health Network, McGill, and we have a site in Italy. And these are the ones that are just onboarding. We have another list of folks that have expressed interest who are now getting ready to be onboarded. Currently we have, and we just started like 6 months ago, so we have about 1,400 patients entered. This is the breakdown. Primary therapy is the majority, 1,300. Salvage, 79. So again, we will collect whatever you put in. So if you want to put in salvage, that's fine. Ablation energy utilized, cryo, about 450 cases. HIFU, 139. Laser, 750. So that's very robust for the laser group. Tulsa is beginning to grow, 17. IRE is going to, I think, really take off. We have support from irreversible electroporation to use all their post-preserved trial data in the focal therapy registry, and this is exactly what we want to do. We want to follow these patients long term, so this is the mechanism to do it. Radiofrequency ablation, 1, et cetera. Patterns of ablation, it's whatever you want to do. So hockey stick, we have 9 reported, hemiablation 168, quadrant 24, and so forth. So you see how this breaks down in terms of some of the numbers. So again, please join and support the Focal Therapy Society. This is something that a lot of us feel very passionate about, that we need to make progress. I think we all struggle with the same items. You heard from the regulatory board this morning at the FDA, some of the things, the concerns that they have. As clinicians, we share those same concerns. I mean, how do we best do this? How do we best treat men? How do we do the follow-up? We want to do this correct and have robust data. I think this is also a way where, you know, if the FDA wanted to, they can look at some of the outcomes, real-world data through this registry. And that was one of the reasons why we put this together. So with that, I just wanted to thank you and ask you for your support going forward. This is a mechanism whereby you can do focal outside of a clinical trial and still be on a registry and meet AUA, EAU, NICE guidelines, whatever guidelines you follow in your part of the world. Thank you.
Thank you, Dr. Plastic. So we'll move on. We have a couple of adjustments in the schedule from what was printed and available online. We have a couple more talks to round out the clinical session this morning, and then we'll break to a little bit more of an extended break than what was scheduled to allow people to get lunch and come back in time for the sessions to start this afternoon. So I'd like to invite next up to talk Dr. Art Rastenhad from Northwell Lenox Hill Hospital, and he'll be giving a talk on how recent advancements in imaging and ablation technologies could influence future focal therapy clinical trial design. So thank you, Dr. Rastenhad.
It's a pleasure to be here. Before I begin, I really like to thank our FDA colleagues. It is, I know, a challenge and a position that you are put in to try to give guidance on these new technologies and implement these things with safety, with the patients in mind. Without your hard work, we would not be able to do this, and I know the position is probably stressful, and the times we've talked on the calls before the meeting, we really appreciate it. Dr. Pinto is extremely helpful, and it's just without this hard work, we're not going to be successful. Dr. Brown and I have chatted. Our real goal is to disseminate information. That's why we're live streaming the event. It'll be recorded. This is not a part of the Focal Therapy Society specific program. We're trying to make this as unbiased and open to all as possible. So again, I want to thank our FDA colleagues for really taking the time to come down. My opportunity today is to talk about new advances in imaging and ablation technologies. How does that fit into future clinical design? I'm going to give more questions and answers, but at least a background to maybe have some unique thoughts and what's the future look like for us. I'm going to go over the new ablation technologies. I'm going to go over the imaging modalities and advancements in that space, as well as what's the impact on clinical trial design. I'm going to try to separate the fact from the fiction. And speaking of that, and how do we get past that? I think it's really with the registry, clinical outcomes, post-market, FDA-approved devices, looking at these outcomes. You can see we have almost 1,700 cases in the registry. The evolution of prostate cancer. Since Dr. Adams first described a unique and most likely rare tumor in prostate cancer in 1853, 150 years later, we're still talking about it, and it's not as rare as he thought. Hugh Hampton Young first described the perineal prostatectomy, as well as known as one of the fathers of urology. Charles Huggins, one of the two winners of the Nobel Prize for Medicine, discovered the association between androgen-driven prostate cancer, aggressiveness, and growth. The 80s were really an amazing time. We had PSA was going through approval that was cleared in 1994. The trust biopsy, the big breakthrough was the needle guide. Oh, we have a spring-loaded needle, now we can hit the prostate. Dr. Walsh with his description of the radical prostatectomy. And the NIH colleagues, which I was a part of that team, with working with Dr. Pinto, Wood, and Choike, coming up with fusion biopsy. They were the first ones to have an FDA clearance for that. And I think it's really led to the complete change in why we're here today. Because if we couldn't see it, we couldn't treat it, and then we couldn't measure that outcome. And I think that's they deserve a lot of credit for really shifting prostate cancer, because everything we knew before is no longer. The Charlie Pound data, looking at recurrence and when it happened, that was based on ultrasound and biopsy only. And that's how the diagnosis was made, under the radical prostatectomy group. And I think the real future is focal therapy. And we're talking about that today, and all you are here because of that. I don't know who said this, but at one of the last FDA meetings, prostate cancer is the chicken nugget of cancers. I don't think it was Dr. Brown, but someone said that, and I took that away from a 2018 meeting. You can freeze it, you can fry it, you can steam it, you can microwave it, you can electrocute it, you can stuff it, and heck, you can cook it. But I make a joke about it because I think that's why we're here. For some reason, my timings, can you clear my timings on my slides, Dan? I'm sorry, I don't know, I think they're copied over. Give me a moment. But that's our challenge, because there's so many different modalities, and we really haven't honed in on one that's better than the other. And I think those are the challenges we're here today. As we mentioned, cryoablation is in that pre-1976 category of approved devices for prostate cancer. The systematic review, this is a lower risk cohort, intermediate and low risk, at an 8 to 25% positive in-field biopsy rate. If you, general urology this month, we'll give it to the NYU colleagues that came out with a paper with a sub 10% negative biopsy rate in the treated lesion. I think these are exciting times. We used to quote patients up to 30% failure rate. You'll look at different technologies, where's that metric? But we're all measuring tissue right now, and hopefully maybe in the future that may change, and that's what we're going to talk about today. HIFU, same thing. I'm trying to make this vendor agnostic, but in a 7,000 patient meta-analysis, the positive infield rate was between 4.5 and 91%. Why is that? There's a lot of different outcomes and how they're measured and how the procedure was done. And we'll talk about more about that a little bit. MRI, ultrasound-guided ablation, there's two vendors in this space, but you're starting to see a trend. They're about a 20 to 30% infield recurrence rate when people are treated, depending on the grade of disease. I don't think it's so much the grade, it's so much the visualization. When we follow up, this is an imaging-based diagnosis now with MRIs, usually as our primary, our initial evaluation for these patients, and then we're treating based on that type of information. a similar group or a similar technology with a similar results. At 24 months, 88% had no evidence of grade group 2 disease or higher within the treated area. I think this is really encouraging, but I think our challenge I'm going to get to is what are we treating and how are you determining what to be treated? LIT, or laser interstitial thermal therapy, they have 11-year data that came out. 60% of their patients were negative in the treatment group. This is a transrectal MRI in gantry laser ablation looking at thermal therapy and changes within the prostate. But at 11 years, you look at, they had a mixed group of patients, but their metastatic free survival rate was 99%. Prostate cancer specific survival rate was also 100%, and overall survival was 98%. What I take away from a lot of these things when I read it is, does all prostate cancer need to be treated? And I think there's a debate with active surveillance. And then does whole gland therapy, if you have so if you have grade group 2 disease, you have a 35% BCR after prostatectomy and radiation. Is that a cure or control? And I think that's why we're here today, to ask those questions. IRE, or irreversible electroporation, if you're not familiar with this, it uses electrical field, and it puts holes inside. Poration puts holes inside cells, and you have apoptosis, and these cells are cleared. They just completed their pivotal study, PRESERVE. I believe accrual was met in July or August of this year, so we're excited to see the results of that. Nanoparticle ablation is using nanoparticles that are injected in through a peripheral IV that circulate and accumulate in tumor cells. And the current publication going out is 45 patients or 44 patients treated. There's a 75% negative biopsy rate at 3 months and 72.7 at 12 months. For disclosure, I am the PI for this study. We have accrued also our 100 patients for our extended trial, and that is in submission at this time. Bipolar radiofrequency ablation, Dr. Taneja was one of the PIs for this, and you're starting to see this consistency. 80% without clinically significant prostate cancer on follow-up biopsy. So we are obtaining a certain level of success in ablating what we say we're going to ablate, but also how did we measure that? How many biopsy cores we're taking from that? These are the things that you have to ask, and how are they staged? The newest kid on the block, this trial just opened, I think, about a month ago. They've treated 4 patients, water vapor ablation. It's a transurethral ablation procedure. It's similar to what people commonly know as Rezum. It's the trade name, but this is a FDA cleared for BPH treatment that has now moved into the oncology space. Again, a similar outcome with biopsy results. 87% showed no Gleason pattern 4 or greater on follow-up biopsy. This is a small cohort, but the study's expanding, and we're really excited to see the results of that. Looking at this, there's a few components I've broken up. We have ultrasound. We've all been using that since the 70s, really, to see the prostate. MRI and molecular imaging is constantly being advanced. Our MRI from 10 years ago is not what our MRI is today for most people. Image fusion and 3D tracking, I think, is the cornerstone of our diagnostic potential. You can't use a cognitive biopsy and then expect to go back to that place after the prostate shape is changed to get back and resample what you treated. If there's cancer in that area, did you undertreat? Did you not treat? Did you undertreat, underplan, or did your modality or you use not adequately treat the patient? These are important things. Spatial recall for image tracking and treatment. I don't think you should be able to have a trial, this is my own personal opinion, but have a trial where you did a biopsy first and not be able to use that original information to rebiopsy. It's about building the care cycle for care. So you have 3D acquisitions, your cores, you go back, you can remerge that and look at that. I think the reason I'm saying it, it's my opinion, but I'm saying it because almost all the vendors that support these CAD and biopsy technologies, it exists today. So I'm not biased towards one specific vendor, but it's something to keep in the back of your head. It's my favorite slide. We went digital to digital in the last 100 years almost. So the 1930s, we did digital biopsies. How many people have done a digital biopsy? Dr. Pinto and I did the same residency program. When the ultrasound machine would break, 2 gloves and a straw is our best friend. And for all the residents and young attendings out there, the straw that comes on the end of the needles, you know, that straw is there for a reason. It's not to protect the needle from you getting stuck taking it out of the package. It's there because that's how we used to use that needle, because we took one glove on, put the next straw, cut it a little shorter so you have enough room, put the straw up to your fingertip, put your second glove on, a little bit of lube and some hope, and you can hopefully hit the prostate, or at least the hard part. I'm kind of shocked that not that many people raised their hands. I only saw like 6 hands. No one else? I'm not that old. I'm feeling a little old now. I didn't get gray hair at least, okay? But so in the 80s, we talked about the ultrasound, the spring-loaded needle gun. That was a revolutionary breakthrough instead of the true cut needle that was used in the past. And now the needle guide and that spring-loaded gun allowed us to really shift how we do this. And then in 2009, Dr. Pinto's group, Choi Keen Wood, was really the first diffusion biopsy. They had the first intellectual property in the field. But that really then had to take a moment that everything we knew from prostate cancer from that point is going to be different if it's an image-based diagnosis. Dr. Emberton spends a lot of time talking about this because he feels it's not always the tissue, it's the imaging. And if it's an imaging-based diagnosis, do we do that for kidney cancer and other cancers? We don't always rebiopsy, but the opportunity of the prostate is actually pretty easy to access. So trial design, I think the inclusion criteria, I think most people in the room would agree, it's a multiparametric MRI of the prostate and an MR or MR ultrasound fusion-guided biopsy. The Focal Therapy Society has come out as suggesting that this is the inclusion criteria for a trial. I think that's helpful to think about that. I mean, as a show of hands, how many people disagree with these types of statements, with the imaging as a surrogate or imaging as a first step and then a targeted biopsy before focal? How many disagree? I'm just doing that because too many people are going to raise their hand. How many people agree with this statement? Most? Because I think if we're going to move to an imaging-based diagnosis for prostate cancer, that's where I believe the field is going. We all have to accept these are the initial steps of that for our design of our future trials. This was a paper that was discussed at the FDA meeting in 2018, which said mostly you need a 1 centimeter margin. I put this up there. I know Lenny. I've talked to the Margolis. When you look at this and you think a centimeter, and the prostate's only 3 centimeters wide, you're almost treating the whole prostate if you had to put a 1 centimeter margin. I think our limitations at the time is the quality of imaging. That's why I put up the T2 so you look at it. Any radiologist says if you look at you want to look at the pictures of the publication before you actually look at their data so you understand what was happening. This is the NYU group, which came to a similar conclusion, but their underestimated size is a little less than what the other study was. And if you look at it, you see a really nicer T2 start to form. And then there's my friends. I was in this project with Dr. Barris-Turkbay, Dr. Choiki. In 2009, we were using PFC 770, instilling this in endorectal balloons on a Philips Achieva, which no one would ever use today, and getting images that are comparable to 2023 using this approach. No longer do we use endorectal balloons for MRI. How many people have endorectal coil MRI still available? It's pretty much been removed from the market because the techs don't want to. You have it still?
We have it too.
Really? All right, so I've stopped fighting the fight with the techs because I hope the amount of the signal noise ratio that we receive is comparable to these results. But how did it pan out? There was some heterogeneity in the field, right? They said it underestimated by 80%, 14 from NYU and the NIH. And this really had to do with the people interpreting what was the goal of their original study. Because when I spoke to the ones that really underestimated, they're like, oh, we're using the biopsy data, not drawing the tumor to the most generous size using each sequence. We wanted them to make sure they found cancer. So getting behind the scenes, you start to learn more about the publications. Now, everyone that knows me knows I always put up this slide. Does MRI quality really matter when you're going to treat a patient? And now we just talked about different conclusions from different sites based on the type of a T2 acquisition they were using at the time. And I would argue that the higher quality MR may give you a better understanding of your disease burden. And if you look at this as a T2 and ADC, clearly you can see this is Gleason 8 on biopsy, downgraded 4+3. But you can see how the small amounts of prostate cancer are now moving through the peripheral zone tissue. And notice the ADC map here underestimates that because you have mixing of the voxel signals of restricted and unrestricted tissue. And I think this is the challenge that we deal with in focal therapy is what is needed to get great results. It's usually how strict is your inclusion criteria, your baseline diagnostic strategy. How was the quality MRI? What type of biopsy is done? How many cores were taken? And these are some of the things that we struggle with when trying to design our trials at our institution, as well as talking to colleagues about trial design. Oh, just one last thing. The AUA guidelines this year, I believe, came out to a minimum of 2 cores from a targeted lesion. And then if you look at our prior transperineal data, it was up to a minimum of 4 cores. So there's some ambiguity here. There's a minimum of 2, of course. But most trials, how many cores were taken to determine that there was no cancer? I literally try to fail when I do a biopsy, 4 or 6 cores. If I'm not happy on my post-ablation and I'm biopsying scar tissue that's really dense, I am sampling outside maybe my ablation zone. I want to know if I failed. If I failed right next to where I said I treated, that's a planning failure. I accidentally undertreated. So that's the challenge. So if you're only taking 2 cores, I don't think it's really going to get you to the best endpoint, because you're really limiting yourself in trying to tell a patient what they actually have. And our goal is to make all this stuff better for them. It's about the patient. So let's try to fail. And if we don't fail, because we've planned ahead so well, I think that's a positive thing from the physician side. This is a paper, 15-year experience. Where's my friend, Dr. Moore? Dr. Moore has been a giant in the field. I saw her come in this morning, but Carolyn Moore, she has won more awards and done more in prostate cancer than most people in their career. She's done a great job. This is their series from Hash, her, and Mark. They've done a great job, and they looked at this. And I think the important thing from their study is that more than 1,000 patients were intermediate or high risk. Mean 7-year follow-up, 69% freedom from failure-free survival after primary HIFU for of course, organ-combined disease. Metastatic-free survival, 100%. They actually also published a recent paper comparing to radical prostatectomy in a matched cohort and showed no difference in the incidence of metastatic disease. And I think that's important. That's where we're trying to get to. We don't have a lot of that data, but that's what's exciting today. As I mentioned about planning, there's a company that has developed an AI technology, basically a color heat map using whole mount histopathology. And you look at it, what's the first thing we do when we look at a study about imaging? Look at the T2. At least you look at it, it's a much nicer T2, and that's how we have to judge some of this. And then when I do a treatment, or when I first see a patient, the MRI is positive, they're going to have a biopsy, I biopsy around the lesion in my templated biopsy to make sure, is the tumor going that far? Do I need to treat that area? So I set myself up to find out if they're really a candidate or not before we even get to discussing focal therapy. And this group is given a heat map to help you predict what your ablation zone should be. I think these are exciting things, and I look forward to seeing stuff like this implemented. But you still have to understand, do you really need it? I don't know. But I know what you do need is good quality imaging and having the foresight to plan your patient at the moment of biopsy if they're going to be a focal candidate, and designing your biopsy template around those areas. I really focus a lot on imaging and quality. This is the idea of PI-qual and what the impact is on, does quality matter? And quality absolutely matters in the acquisition of the MRI and the outcome. And how does it matter? This was a large multi-center study looking at PI-qual. If it was less than 3, of course, this makes sense. Poor quality imaging, you find less lesions. You're unable to assess if there's extra prostatic extension, which then would drive the number of PI-RADS 5s you see, because that's the way the scoring system is developed, right? If you miss EPE, of course it's going to be a 4 if it's less than 1.5 centimeters. So the way the kind of their conclusion, it kind of conflates one with the other. It's not you're missing PI-RADS 5, you're just missing EPE most likely driving that, or it undersized the size of the lesion. I take a lot of time with this, and I talk about image quality a lot because I think this really impacts our patient outcomes and care and planning for our procedures. So that's good. There's a great study, they did that. But then, what are the limitations for MRI? This is another study that just looked at an enriched population of patients with prostate cancer, just comparing where PSMA, PET CT fit in. In the United States, this is an expensive test. In Australia, you can get this for $1,200 or something. Very cheap. It's available everywhere. It was many years before it was available in the United States. And they really found that there was no difference between the two cohorts, but when you put it together, you can see one missed 10%, one missed 8% of the patients with Gleason grade group 2 or higher. Dr. Pinto and his colleagues, Dr. Rothberg, also went through a literature review and came to the same conclusions, and it kind of makes sense. I think our challenge today in the focal therapy space is being able to obtain a PET CT for a patient that doesn't have grade group 4, cribriform, or high-risk features, or PSA greater than 20, because that's not the indication for that test in the United States. It's helpful if you want to rule out, extra prostatic disease. And to speak to this for one moment, so if you have an MRI with a lymph node that's 8 millimeters, study's done. That is not positive for metastatic prostate cancer in the known setting of Gleason 8 prostate cancer, because most of the time, less than 1 centimeter, they hedge, it's indeterminate. But you can have a 4 millimeter PET positive lymph node very easily, so it does change the stage of the disease when we're using advanced imaging. This was another study looking at, can we move away from the prostate biopsy? Okay, take a deep breath. It is an extremely high-risk group of patients that they did this in, and it's a German study. Someone sent this to me and was like, we should be doing this here. I'm like, there is no way we're going to be operating on patients without tissue at this time, at least it is for initial diagnosis. But this was done, and they felt that this could be feasible in the future. I'm just putting that out there for a discussion point. I am not endorsing this at all. But again, where in focal therapy does PSMA PET CT fit in? This was an abstract looking at the positive predictive value in patients who failed focal therapy, and it was 89% in the PET group and 88% in the MRI group. So imaging was able to detect this with some accuracy. Now looking at the paradigm shift, we heard about where we were. Our current paradigm before considering focal therapy is still investigational. John Coleman put a slide like this up. I've edited this over the last 5 years. But we were in a paradigm where there was no focal. It was active surveillance and then radical therapies. Then we had a paradigm where it was looking at low-risk prostate cancer. I think the problem is people have talked about TCAD. I'll just at least mention it. There was more complications in the treatment than there was a benefit for the patient to be treated. So I think that's the challenge. So people could eliminate going on to whole gland therapy, but they had a very high complication rate. And I think that just kind of summarizes what the problem was when that study was designed. Our current focal paradigm, we are moving away from low-risk prostate cancer. Some low-risk prostate cancer, high volume, can behave like grade group 2 in some publications. We're focused in that intermediate risk cancer group. We've talked about trying to substratify the unfavorable versus the favorable. That's kind of where we are. Some people have published on the high-risk cohorts as treating a focal grade group 8 disease or grade group 4 disease, and if the rest of prostate looks pristine and there's one spot, there's the possibility of that. That is done on trial. And when you think about what's happening is that as we switch to an image-based diagnostic pathway, you're seeing it, you can treat it, and then you can follow it. And that's where we're kind of trying to change this whole cycle of how we looked at prostate cancer and where is it going to take us. So trial design, I know this was touched on this morning, but metastatic-free survival, I think the FDA team, you mentioned a 3-year window or a 5-year window for the incidence. I think that's how I interpreted that. This trial had to be 5 years. Maybe I heard that wrong. We can discuss it afterwards. I didn't know what's a reasonable time point. Upgrading on tissue biopsies, typically biopsies done in the 6-month to 1-year mark. Will imaging be an alternative in the future? If so, what types? And looking from freedom from treatment, whole gland treatment, is that another metric? And those are the important things that we're going to discuss today in our open forum and questions. I think prostate cancer is or has shifted to an image-based diagnostic pathway. Combining the modalities I mentioned, is that a possibility? I don't know. Can a biomarker maybe overcome some of the associated limitations of PSA, looking at diagnosis and or outcomes? If you have a positive biomarker pre-op and then post-op the biomarker is negative for grade group 2 disease or higher, would that be something that we would look at? That I don't believe has been done on a large scale. And that's where I'm going to leave you for this morning with some thoughts. It's really not meant for me to tell you what trial design should be, but to have a discussion about that. Thank you.
All right, so thank you, Dr. Rastenhead. So for our next talk, we will invite Dr. Howard Parnes up. He is the Chief of Prostate and GU Cancer in the Division of Cancer Prevention at the National Cancer Institute, and he'll be giving a talk this morning on prostate cancer focal therapy, providing context for clinical trial endpoints.
Is this mic on? Now it is. Thanks, good to be here. I'm Howard Parnes. As just said, I'm Chief of the Prostate and Urologic Cancer Research Group in the Division of Cancer Prevention at the NCI. And as such, active surveillance and ways of delaying or preventing progression of prostate cancer has been a main focus of mine since I started working with Peter Pinto close to 15 years ago in our multidisciplinary clinic. And I've gotten to hear a lot of discussions of focal therapy as we see folks in the clinic, but haven't really delved into this very deeply until more recently. And so it's a pleasure to be here and have a chance to contribute to these wonderful presentations we've heard this morning. I don't know that I'll have that much to add, but I thought it might be useful to provide some context for focal therapy, particularly as an adjunct to active surveillance to delay or prevent progression to radical therapy, and provide some clinical context to inform the discussion of trial endpoints for focal therapy studies. No disclosures. So by way of review, as we all know, the rapid increase in prostate cancer incidence. There is a pointer up here, isn't there?
There's on the clicker.
Oh, down here. Does that work? Well, the rapid increase in prostate cancer incidence that followed the widespread adoption of PSA screening in the late 80s, and the fact that the mortality rates remained relatively stable, despite the fact that many more men were being cured by radical therapy, led to the establishment of large cohorts to assess the feasibility of selective delayed intervention, which has come to be known as active surveillance. That's okay. In the mid-90s, active surveillance cohorts were initiated in Toronto and at Hopkins, and of course at other places as well, UCSF in particular. The Toronto cohort included men with low risk and intermediate risk disease, and surveillance biopsies were performed every 3 to 4 years. The Hopkins cohort enrolled men with low risk and very low risk prostate cancer and performed annual surveillance biopsies. With follow-up out to 20 years, the combined rate of metastasis and prostate cancer mortality in the Toronto active surveillance cohort was 3%. About 25% of patients switched to radical therapy during the first 5 years of surveillance, although only about a third of these men came off surveillance due to increased tumor grade on biopsy. In the Hopkins cohort, the combined incidence of cancer-specific mortality and metastasis was only 0.1% at 15 years, and it's shown here in the blue curve. About a third of patients switched to definitive therapy during the first 5 years of surveillance, and here about 2/3 came off due to grade progression. As in Toronto, the risk of reclassification was highest during the first 5 years, suggesting that initial misclassification rather than true progression was responsible for a substantial portion of the grade progression. Now, the as we know, and the reason we're all here has to do with the advent of MRI and the ability to perform MRI-guided biopsies, which has changed the prostate cancer landscape in several ways. First, it has substantially reduced the grade misclassification. It has also contributed to further stage migration, the initial wave, of course, being the widespread adoption of PSA screening. But it's contributed to the further stage migration by enhancing the detection of small volume, higher grade disease. MRI, of course, has led to the rapid growth of focal therapy by enabling detection of discrete foci of disease amenable to ablation. However, active surveillance, which is now endorsed by the NCCN for both low risk. Oh, okay.
Because it's not showing up, so just follow along.
What's not showing up?
So this computer isn't showing up for some reason. So just
Okay.
Just double check the slides, Dr. Barnes. Just make sure that it's on the right one.
Here.
you're on 15. You're on 3 more.
Oh, so you guys didn't see these?
Yeah, no. It's causing trouble. For some reason, your computer's not connecting.
Oh, okay.
So just have your notes here.
Yeah.
And then use this guy, and it'll advance your slides for you.
Oh, all right, fine.
Big button.
You'll to advance?
Yeah. Looks like that clicker's working now.
Oh, okay. All righty. Well, as I was saying, the MRI has changed the landscape by decreasing grade misclassification, furthering stage migration, and allowing the growth, rapid growth of focal therapy. The NCCN has endorsed the use of surveillance for both low risk and favorable intermediate risk prostate cancer, but does not recommend focal therapy for the primary treatment of localized disease due to the lack of long-term data comparing these treatments to radiation or prostatectomy. And I think the same can be said about the lack of long-term data to assess the utility of focal therapy in patients on active surveillance. There we go. Okay. A recent systematic review identified 72 studies using 8 different energy sources to deliver focal therapy to nearly 6,000 patients. But only 3 of these studies were designed to assess efficacy. One of these was a randomized controlled trial that we've heard mentioned several times this morning of photodynamic therapy versus active surveillance. This trial was conducted in men with low-risk prostate cancer confined to one lobe of the gland, and they were treated with hemiablation using 2CAD as a photosensitizer. Follow-up biopsies were performed at 12 and 24 months, and the primary endpoints were grade progression and absence of cancer on the 24-month follow-up biopsy. Secondary endpoint was progression to definitive therapy, but that was routinely done for just minor changes in grade, as we'll see in a second. Okay. The trial met both of its co-primary endpoints with a 30% absolute decrease in grade progression and a 35% greater likelihood of having a negative biopsy at 24 months for the PDT arm. However, a follow-up paper a year later demonstrated that the reduction in grade group progression was largely driven by a decrease in progression from grade group 1 to grade group 2, so 8.7% versus 27%. However, progression to grade group 2 is not a particularly good surrogate for a meaningful long-term endpoint, and really no longer an automatic trigger for radical therapy as we become more comfortable keeping men with low-volume grade group 2 disease on surveillance. In addition, PDT hemiablation was associated with an increase in grade 1 and grade 2 sexual and urinary side effects, as shown here. In terms of efficacy trials, there were also 2 non-randomized comparisons of focal therapy and prostatectomy. And although I'm mostly talking about active surveillance here, I think these are worth mentioning because they do tell us something about the side effect profile of focal therapy. So one of these trials, okay, two non-randomized comparisons of focal therapy and RP. Here we go. One study included men treated with either cryo or HIFU, while the other was of IRE. The study populations were similar, although there was more grade group 2 and grade group 3 disease in the IRE study. In the cryo-HIFU study, an entire lobe of the gland was treated, whereas the IRE study used MRI guidance to deliver unifocal ablation. The residual infield cancer rate was 29 and 30% in the 2 arms, respectively, and the salvage treatment rates were 21% at 46 months and 14% at 12 months. Both studies reported better erectile function and pad-free continence at 12 months with focal therapy, although the relatively short follow-up may have led to an overestimation of the harms of RP, as these side effects often do improve beyond 12 months. And notably, IRE was not associated with a significant improvement in epic urinary summary scores, despite the improvement in the pad-free continence. And although functional outcomes were better for IRE than RP, IRE was associated, I keep forgetting to do that, IRE was associated with a decrement in both urinary and sexual function. Better than RP, but nevertheless, you see the curves take a dip after treatment. The top curves show men who had full function. The top curve is all patients, and the lower curves is men who had full function at the outset. And similarly, the cryo-HIFU study was associated with a decrease in sexual function. Again, better than RP, but you see, there we go. Better than RP if we look at on the right-hand side, but yet a decrement following treatment. So these therapies are not totally benign. Finally, there was one retrospective analysis that's already been mentioned today, Dr. Moore's study of oncological outcomes in men treated with either focal or hemiablative HIFU. This analysis of over 1,000 men, 63% were grade group 2, 17% grade group 3 or 4. What's going on here? Did you not see these slides? You did? I thought so. You saw this? Mike, how do we do? Oh, there we go. Okay. At 96 months, as we already heard this morning, overall survival was 97%, upper left panel. About half had persistent grade group 2 disease, the upper right panel. About half had received further treatment, and 19% had undergone radical therapy, which is a lower rate of a lower rate of radical therapy than previously reported for active surveillance series. This is a pretty good review article of active surveillance series. In the middle, we see the data from Toronto, Dr. Klotz, and then taking a closer look at that. If we compare this to the Toronto series, in which 36%, nearly twice as many men, had switched to radical therapy at 10 years, compared to the 19% at 8 years. However, as we all know, these cross-study comparisons are extremely hazardous and can be very misleading because of differences between the cohorts, the follow-up, the triggers for intervention, which all goes to underscore the need for prospective randomized trials. And I think these trials should focus on both GU, GI functional outcomes and oncologic outcomes, the key functional outcomes, GU, GI sexual side effects, as well as patient-reported quality of life using validated tools such as EPIC, et cetera. The oncologic outcomes will need to include surrogate endpoints such as grade group progression, time to definitive therapy, and retreatment rates, given the very large sample sizes and long follow-up that will be required to obtain meaningful data on rates of metastasis and disease-specific mortality. And so in summary, questions remain as to whether focal therapy provides better quality of life than radical therapy, and whether focal therapy provides better oncologic outcomes than active surveillance. And as we embark on trials to answer these questions, we will need to be mindful of the large and growing number of focal therapy techniques. Otherwise, we won't know if findings regarding any particular type of focal therapy are generalizable to alternative focal therapy approaches. So thank you for your attention.
All right. Thank you, Dr. Parnes. So what I'd like to do now is we'll have just a little bit of a panel and question discussion session to review some of the content that was presented here from the clinical aspect. And at this point, I'd like to invite Dr. Pinto to come back up, as well as Dr. Antonino and Dr. Suzman from the FDA, and Dr. Brown, if he's here as well. We'll make some room on the panel. I'll step aside. And if any folks have any questions based on what was just presented or stuff that was presented earlier this morning from our colleagues at the FDA, I invite you to ask those questions at this point. So.
So a question and maybe just a perspective. So I think there's a real emphasis, and maybe somewhat misguided, to do randomized studies. So what I like to do is look at PLCO, right? And PLCO said we shouldn't screen, right? A randomized study. How many in this room screen? Okay. So let's look at PCPT. It said we should prevent prostate cancer progression with a 5-ARI. How many do that? We got one. The other randomized study, Pivot, said we shouldn't do radicals. How many do radicals? So there you go. That's the impact of randomized studies in prostate cancer. And I'm convinced if you did a randomized study, however which way you want to do it, with active surveillance or the focal therapy, which was done, and the FDA sort of turned the other cheek on that, or you do it with focal therapy versus radical prostatectomy, if you can actually randomize those patients, I don't think it's going to have a big impact, just as all the randomized studies that have been done in prostate cancer have really not done so. So I think we may be best off with quality data that isn't randomized, but quality in terms of enrollment criteria, treatment, and endpoints, as far as really focusing on randomization, because it really hasn't done much for prostate cancer so far in this lower intermediate risk of patients.
Dr. Lepore, you just can't say that and then walk away. I mean, that's very, I mean, that is real, I mean, that's a good question to start with. I mean, that's a good point. So, because the problem is, as you walk away, then they're not going to answer your question, right? So let's stay here. Come on, let's think about this. We have a little time.
I was asking what the question was.
The gift of this session is we have a little time.
The question is the emphasis on randomization studies, because that's going to encumber the enrollment, and it's not going to have an impact, just like all the randomized studies that have been done. And maybe what we need is high-quality data as opposed to focusing on randomization. So my question is, do we still need randomization?
Yeah. Yeah, I kind of couldn't disagree more with that stance. And it reminds me of...
On the basis of what?
Well, it reminds me of the early in my career, more than once did I hear Dr. Walsh from the podium talk about the need for a blue ribbon registry. We don't need randomized trials.
I'm a clone of his.
Yeah, and we don't. And what we needed the randomized trials. We needed SPG4. We needed Pivot. We needed Protect, or we would not have made the progress that we've made.
Well, Pivot said we shouldn't do radicals, and we do. PCPT said the primary endpoint was to prevent prostate cancer. It met its endpoint, and we didn't. And PCP and the PLCO said we shouldn't screen.
Okay, so we could have a
the problem is in the methodology, and I'm convinced that if you do a randomized study, it will encumber enrollment. And once you go through it, we're all going to sit there and do what we want anyway.
Yeah, I think that's a straw man argument is to enumerate.
Well, give me another study in this space where it's impacted.
You've enumerated 3 trials and said we didn't do what the trial said.
I'll give you the 4th. The one was the act. No, I'm saying we do, and it doesn't have impact. Yeah. And if you get hung up on doing randomization, it's going to impact our ability to have quality data. And at the end of the day, at least we're not talking about, with castrate-resistant prostate cancer. We're talking about in this space with lower intermediate risk disease. And I'm just looking for a randomized study that's really impacted what we do.
Well,
or to the reverse. In terms of the primary endpoint, was reached, and everyone just sort of said, okay, well, I'm going to take out prostates, I'm going to screen, and I'm not going to use I'm not going to use 5-ARIs.
It is
provocative.
It is
question,
certainly
but
true
it's
that
real.
non-randomized trials can be incredibly useful, and I presented 2 of them today. The Toronto active surveillance cohort and the Hopkins in a non-randomized trial very strongly supported the safety of active surveillance so it doesn't mean that you can't answer questions with non-randomized trials, but I just don't think you can dispense with them by saying that we don't follow the results. Each one of the trials you mentioned is worthy of a discussion for how those results have impacted practice, and we didn't just throw them out. They all were important in the context of other trials. Pivot said it didn't work, and then Protect. As we went from SPCG4 to Pivot to Protect, what we saw was a diminishing return for radical therapy as patients got diagnosed earlier and earlier over time. The original Swedish study with 23 years of follow-up shows a 12% prostate cancer mortality benefit and an overall mortality benefit. That was not seen in Pivot, but the same relative risk reduction, about 0.5, 0.6, was seen in Pivot because we had a less advanced population. Protect, you see nothing. You have 99% survival at 10 years, but you do see a little metastasis signal.
So if we look at the Scandinavian trial,
we learned a lot from those randomized trials.
Yeah. So what did we learn? We learned from the Scandinavian trial that 31% of men died if they were on watchful waiting, and 20% died if they had a radical. Right. So you took 9 radicals to treat to save a mortality. And metastases, you have to do 6. So I think we're so concerned about our focal therapy and all the harms that we might do. But if we take the Scandinavian trial, what we learn, there's a lot of people that die of prostate cancer if we don't treat them, if we treat them, and there's many that don't. So I would sort of use those studies to be a fairly compelling argument for focal therapy, having taken 5,000 prostates out.
Dr. Lepore, just in defense of the impact of these randomized trials, the utilization of prostatectomy and whole gland therapy went through a huge change in impact of this. I would say I've never removed one low-risk prostate cancer, ever, right? And so I would pose that as new generations of prostatectomists have come out, that's never happened.
Yeah, but that wasn't based on the randomized study.
No, it was based on the influence of this data coming out during the formative years of my practice, right? And so That wasn't true for the people that trained me who were excellent prostatectomists. They removed a lot of low-risk disease. And the lessons learned aren't black and white, right? It's who is best served. And in a randomized data set, there's going to be winners and losers. And you can certainly lose signal if you don't enroll correctly, right? So to say we are absolutely unchanged by the data, level 1 evidence, we are going to be most influenced by level 1 evidence by studying. And just because you get no signal, there's type 1 errors and type 2 errors for every trial design. So
we don't do radicals because of the open label active surveillance studies, right? That's what really, I think had its greatest impact, right?
Yeah, I would say that the practitioners that do the most amount of screening, who are internal medicine physicians and family practitioners, abandoned screening because they saw it fall apart when therapy was applied too widely, right? Was that beneficial? No, it was not.
But that wasn't a randomized study that drove that. That was a Toronto and Hopkins data that drove that.
Well, true, but I would...
I'm just saying, I think there's, I'm not making an argument against good quality data. We're probably not going to get that data if we focus on randomization.
Sure.
That's really the point that I'm making.
Dead silence.
So can we? No. So let's hear about it. So Mark, Dan, any comments on this? I'm not going to let this go. This is good.
Yeah, I mean, I'll say, again, the FDA doesn't regulate practice of medicine, but we do regulate the products. So I think you're going to have difficulty demonstrating the impact of your product in this population without a randomized trial. And the randomization helps to mitigate confounding data, that both known confounders and unknown confounders that can influence things like time to progression events or whatever other endpoint you're using. I think we're going to be flying blind without.
But I think that was done in the 2CAD study, but then there was an objection to the design, right? Because, I mean, that was a pretty compelling endpoint, right?
Yeah, I mean, I think the concerns around the 2CAD study, I think, were around really demonstrating that benefit. That again, I agree. It had some of the progression, Gleason progression data. What we're really focusing on for the clinical benefit is not just that clinical benefit, not just that time to regression data, but really demonstrating that there's a clinical benefit in terms of reduction of morbidity, reduction of equivalence in further oncologic endpoints. And I think there were trial design flaws that hinder the interpretability of that trial, not just the fact that it met its primary endpoint. I think that is only one consideration among others.
But I think we see that. Look, we all look back at PLCO and said it was gross contamination, right? But it was a randomized study. But I think what happens is and look, we can look at Pivot, right? They were supposed to have 2,000 patients. They had 700 patients. It was in the VA. Half of them died because of their comorbidities. So that was a terrible study. But it was a randomized study. And I think many times if you try to do a randomized study in this space, we're going to meet the same fate. And look, I don't like the idea, the decision about whether you go on to radical prostatectomy in the 2CAD versus no treatment. The problem is that was a totally subjective decision, who got a radical and who didn't. Right? So these are all the methodologic problems that it's going to be very hard to tease out. And my big concern is that we're going to still have a lot of I think the focus should be on really quality data that can inform decision making, as opposed to focusing on trying to do randomized trials, because you're going to get a highly biased cohort, because people already have a pretty sense bias of who they're going to enroll into these trials. It's just a thought. I think what we really need is really high quality data with well-defined enrollment criteria, well-defined oncologic outcomes, and I think that's where we should put put our emphasis in.
Dr. Antonino, any comments on the utility of that kind of data in terms of how it might influence things from the FDA standpoint?
You know, there's been a paradigm of randomized controlled trials for medical device approvals or clearances for quite some time. You know, we certainly consider this, you know, as we think more about it. But at this time, I think, you know, for reasons that come to mind to me are, you know, the bias, you know, the selection of, you know, treatment versus a control, you know, how that, you know, impacts the outcome of and usability of the data, how it relates back to the SAP plan. I mean, all of these considerations historically have, you know, led us to use randomized controlled trials. So, you know, again, considered, understood. I hear your points.
You know, and my concern is in the absence of this data, you know, the train's going to leave the station, and it's going to eventually get to the point where quality data, you know, everyone's already just, you know, treating patients. And again, we have the registry, which I think is critical, well, which can be, but it's such a different range of who's in it and how you're following the patients. And it's just a matter of having a cohort of patients that's thoughtfully followed, that we can inform, you know. Well, I think what we need is, how do we inform enrollment? How do we inform the appropriate oncologic follow-up? And then are we ultimately doing good for our patients with prostate cancer?
Thank you. Before Dr. Moore comes from the future, and this entire space, we all are in a specific bubble in our heads of where our thought process is. So if you started fusion biopsies and MRI in early 2000, you have a different perspective on reality. And we saw that with people getting into fusion biopsy, asking similar clinical questions 7 years after we wrote the original paper that answered that question. And the UK and Dr. Moore's group, Dr. Moore is a giant. And I said that she wasn't here, but she's from the future, because they are way ahead of the US on the thought process on focal therapy and where they are. And I wanted her to give her basic perspective, and then how they've come to these conclusions. Because in the UK, they do have a multidisciplinary approach that's different than the United States, with true multidisciplinary input and approvals and recommendations. And this bleeds over not into cancer, but into benign treatments and diseases. And I think the challenge is in the US, where we are and where we are on our thought curve, we're maybe 5 years behind if you haven't been in this space since early 2000. I believe Dr. Moore started early 2000. I want to date her.
I'm going to sit down, but I'll congratulate Dr. Moore. And I would say we've learned an awful lot from that quality study. And I think that's sort of the point that I'm making. Is we need more quality data like we have from the group at UCL, and I think that's going to have a greater impact in informing what we do than focusing and getting sidetracked with randomization.
Listen, before Dr. Morris says anything, because I want to hear about the future also, I just want to make a point. So we have a lot of young trainees in this society. This is a young society. We heard from Dr. Verganti. In his practice, he's been out in practice now as an attending for many years, that, you know, the landscape has shifted appropriately. I think Dr. Laporte's comment, what I take away from what he said is, we can learn from a non-randomized trial, and we can learn how to better manage patients with a non-randomized trial. And Dr. Parnes' lecture about the surveillance data that came from Toronto and Hopkins and other places is not a randomized trial, and we've helped out patients. I think nobody would disagree that by operating on fewer low-grade, low-volume prostate cancer is a health benefit to our patients. And at the time when that was the operation du jour, it was because we didn't know better. And every surgeon who took out a low-volume, low-grade prostate cancer at that time said, this is saving a life. And maybe years from now, focal therapy will be in that niche, but we can learn from both. What I want to just remind the audience is, you know, we're looking and we'll reach out to you about subcommittees for the society, opportunities to answer these things, and our partnership with the FDA, people like Howard Parnes, who is a lifelong civil servant in the support of prostate cancer patients in the cancer prevention site, will also be part of this, and it's work to be done. So what we're hearing today is that we need to do more work in this field. That's all. Great question. Thank you. The future.
I feel nervous to speak now from the future. But I guess I am in a really different environment. And I think there have been a lot of valid points made. So you do need, I think a randomized trial is not enough to change practice if the people who are needed to change practice don't believe in the results. So we did the precision study, standard biopsy, or MRI on a target. We didn't do it because we wondered what the answer was. We had no doubt, and we'd been doing MR targeted biopsies for years. But what we saw was that the world didn't believe that. So how could we try to change practice in the world and get it into the UK guidelines? So we had a very thoughtful group of people come together to co-design the trial. So it's really crucial that you get the people who will implement the practice to be part of the design. Otherwise, you design a trial, dare I say, I'm sure this is a closed room, Chatham House rules, the TCAD trial, I think, was in the wrong population. 3 + 3, who cares? You know, so in the UK, where we did all the early work, it's approved by the EMEA, so the drug can be used. But in the UK, different to the US, the NICE does regulate medical practice. So unless NICE approves it, you can't do it. So you're allowed to use the drug because of the EMEA, but you can't use it in UK practice because NICE don't approve it for medical practice because the results are in Gleason 3 + 3, which we don't treat. So one of the other sort of aspects is the other bits of regulation, the MDT. So people, if you take out a prostate for Gleason 3 + 3 in the UK, then the head of the National Prostate Cancer Audit writes to your chief executive to say, this year you took out 5 prostates for this reason. And you say, oh, it was undersampling, we thought it was this, severe psychological distress. But when you know you've got to write a letter which gets published in the national audit, you don't just sign somebody up for it, you change it. So changing the other mechanisms also helps. But I think, you know, there's lots of good things here, bringing together the community who can do the trial. So this is the community that should design the trial, that will then be set to the FDA and will help to influence practice. That's sort of the way that we do it in the UK. We do have a randomized trial in the UK, which is open, called PART. So it's sort of the Protect team, so Freddie Hamdi, and our team at UCL and others across the country looking to randomize men between focal treatment and radical prostatectomy. And it will work best, and the pilot showed that it works best in the places where they don't yet offer it, a bit like with Precision. We weren't the highest enrollers of Precision, but we enrolled people. But the places where they couldn't offer it without it, it was helpful. So working out where you might show your results versus where you might use your results is another important factor. And then again in the UK, there's a big emphasis on patients being involved in trial design. So something to consider when you're designing the trials.
Before you walk away, I think there's that publication where you did look at a cohort comparing focal to radical prostatectomy, the incidence of prostate cancer-specific mortality, and the incidence of metastatic disease, which were some of our secondary endpoints. Can you speak to those results and then how you use those results now change practice? Because I believe they kind of supported or may support the use of focal with less morbidity with similar oncologic outcomes.
So I think you might be referring to the 7-year outcome analysis, the propensity matched analysis. Was that the one? Yeah. So that was HIFU or cryotherapy from the heat and ice registries in the UK. Propensity matched for Gleason grade, MRI findings, cancer volume. Again, I think we've got the whole of risk stratification wrong because we don't properly have volume and MR in there. But and it essentially said so we took the need for additional treatment as your sort of a bar, and it's Hashim Ahmed who led the work, but I'm obviously familiar with it and part of it. And it showed that there was no difference in those retreatment rates, and I've got some slides on it that I'll show. So sort of working out the parameters of what you're avoiding is also helpful.
So excellent points, Professor Moore. So I turn to the panel. We talk about the importance of real-world data and randomized trials versus registries and tailoring it such that the results and the endpoints that are studied are going to be meaningful such that the people in this room, the people who are going to be guiding treatment and offering this to patients are convinced at the efficacy and safety. Comments from the panel, what are the most clinically meaningful things you guys consider for those endpoints? What is the data that's going to convince Dr. Suzman, Dr. Antonino, and the rest of their FDA colleagues of safety and efficacy? And then to our clinicians here, what's the data and what are the endpoints that are convincing you from a clinical standpoint in terms of implementation of these modalities?
Sure. So I think we sort of discussed a lot of it. It's clearly demonstrating in the active surveillance, assuming you're looking at a lower, relatively lower risk population where the alternative is active surveillance, that you're clinically benefiting these patients, whether and demonstrating that via likely a prospective randomized trial. I think we've considered other types of data in the past, but I think that's probably the cleanest way to do it is a prospective randomized trial where you're clearly demonstrating that you're both impacting an objective primary endpoint that would trigger radical therapy, and that you're demonstrating that it's less toxic than the alternative, and maintaining equivalent oncologic outcomes.
But the one question I always have is why compare it to active surveillance? Why aren't we like Dr. Moore and looked at compared to whole gland therapy as in radiation or surgery? And it appears from what I've seen this morning that grade group 3 has been taken off the table in trial design for device development as part of an inclusion criteria because you feel it's doing undue harm to a patient that could have whole gland therapy. That's what I took away today. But the number of events that occur in that grade group 3 group would be meaningful because then we would have a better answer because they would occur at a higher frequency compared to grade group 2. And if the group in the room is really focused on the idea that we can ablate and we can control cancer, I see discounting that subgroup of patients may not give us the answer that we really want because we can live or die by these clinical choices, and that's the trial design question. I want to put it back to you is that grade group 3 group, that unfavorable intermediate risk group, not based on T2, B, or C, but really PSA less than 20 grade group 3 disease. That's one, those have more than one risk factor for that by the NCCN. Why would they not be eligible from your view as candidates for focal therapy trials?
I'll let Mark speak from the device side. I don't think from the drug side. I think we wouldn't necessarily be opposed to it. I think it's a question of is there adequate safety and efficacy data to justify it, and what would your control arm be, and then what would the trial design and endpoint be? I think I see a lot of challenges in comparing it to whole gland therapy because what's your trial design going to be? It's unlikely you're going to be superior to a whole gland therapy on your oncologic efficacy endpoints. So at that point, you're sort of talking non-inferiority, and that comes with a lot of challenges.
I wish Mike was here to answer some of this. But yeah, I think from the outset, we look at it, based upon a management versus curative claim. I think that's the first consideration. And then, if you are, going for a curative claim for a GG3 group, there's concern with the ethical implication of not, treating them effectively, and that comes with, longer term, follow-up considerations. So I think a lot of that was covered in Mike's conversation, Mike's talk. But I think again, as Dan said, we're open to it if there's scientific justification that these patients would be treated ethically and there's proper safety, catch falls in place.
Dr. Siddiqui.
Actually, Naj, one thing, I'm sorry. I'm going to steal this for a second.
Cut off by Dr. Pinto.
As one of my favorite fellows, I can do this to him. So listen, this goes back to Dr. Laporte's first question, right? So what changes medical practice? How many people have had a person with high-grade bladder cancer that we have evidence, level 1 evidence that they should get preoperative chemotherapy, but we say, let's take them to surgery first, let's get this over with. I know the data's there, but we're going to practice this way. I've been doing this for 20 years. I'm going to take out your bladder first, and then we'll get you over to medical oncology, see what the pathology shows, and we'll go from there. Now, what Caroline Moore said is that they regulate medical practice, they being the government. I wasn't sure what NICE was, Caroline, but I looked it up just now, and if I didn't know, maybe others don't know, but NICE is the National Institute for Health and Care Excellence. Is that correct? Okay. And so we heard from our FDA colleagues that they do not regulate medical practice. The government in this country does not regulate medical practice, but it sounds like in the UK, the government regulates medical practice. So gee whiz, how different. Now she's from the future, but she's from a very different future than in this country, because if in this country people regulated your practice, Dr. Lepore, you can't do this surgery because the trial didn't say you could do that operation. And I'm not saying which is better or worse, maybe the UK is better, but we have to work with the rules of our game. And in this game, in this country, the rules are we have to change practice behavior by doing the best we can, and sometimes even the best trials still don't change practice. But I think all the young colleagues who are in this room, as Dr. Vaganti, will slowly adapt. But that's why I wanted to stress that point. The government in the UK is regulating medical practice. In this country, they do not.
But just to realize that to support NICE a little bit, 2015, I believe they came out with MRI in the prior negative biopsy group and showed that biomarkers from the serum did not impact clinical decision to have a biopsy or not. So they're ahead of the curve. Again, another touchy subject, not for this meeting, but prostate artery embolization. The AUA has come out against it. There's only 2 AUA members that do the procedure. They're protecting their membership by that because the NICE guidelines also through a multi-D group, it's true multi-D and input, came out that that's a reasonable and treatment with efficacy that is demonstrated and should be an option for patients. And that's our challenge. Which future is it? But their future seems to be taking to all players, not just one group's point of view, and they don't have an organization like the AUA that says we're protecting our memberships over advancing care. You have an inherent bias built in with organizations too. We're biased. We're called the Focal Therapy Society. Like right as you walk in the door, everything written it calls about focal therapy. We are a very biased group of people. We flew from around the world to come to this meeting. So, Dr. Moore?
So yeah, I like working in the UK. There are some good things to the system. So in 2014, it was the 2014 NICE guidelines that said we could do. So what they actually said was that you can do an MRI after a negative biopsy because you might have missed something, and you must do an MRI after a positive biopsy for staging. So we wrote, and I wrote with a number of different hats to say, so if somebody has a biopsy and they're fit for radical treatment, we're going to do an MRI. Why don't we do the MRI first, and then we get a better MRI? And they didn't agree to it at that point, but they did in 2018 after we'd done precision. So we did precision, and a lot of work from sort of other people and our charity. So the Prostate Cancer UK did a lot of lobbying at different levels to sort of get the conversation on the government timetable, that kind of thing. And that is part of the mechanism. So what it means is, I think the best example of practice really is Stampede. So Stampede is our metastatic prostate cancer trial. It's an ongoing trial where the things that don't work get chucked out and new things that might be good get included. And the mechanism is such, I think it took 3 weeks from the trial results being published for it to be NICE approved practice for a drug, not for a procedure, which is harder to sort of implement. But I like the UK mechanisms on the whole, when I'm not annoyed with them.
And Art, listen, this is the thing. It's a healthy bias. The FTS, this society is a healthy bias, right? But we are also open-minded. Howard Parnes, medical oncologist, who's done so many randomized trials in this space of prostate cancer, he's here, right? We have the FDA. So we're not, speaking to ourselves. We were inclusive of other specialties. And maybe the UK is correct, but I think we have to remind ourselves, what are the rules? We're not going to see a day where the government in the US regulates medical practice. I doubt it. So let's go ahead and know what we're dealing with and then work within the system, or we move to the UK. And Carolina, I'll come over.
I just wanted to add one comment, because we're talking about the stakeholders. And Pinto works for the government, so he can do whatever he wants. It's always, added to the the national bill.
Every patient is on the clinical trial, so. Yeah.
Right. Everything's clinical trial. So I just want to recognize that Dr. Moore's group has done excellent work, level 1 data, all the rest. It clearly states MRI before biopsy. We should be going to targeted biopsy. And yet in our state, I can't get the insurance carriers to approve the MRI first. So what really data are we looking at that changes practice? Here's randomized trial data. We can't do it because we have another invested interest, the insurance carrier, who won't approve it. So what do we do with all this data? How do we?
Actually, I have a question about that. So I wrote so obviously most of my work is NHS. I wrote the Bupa pathway, so the biggest insurer for private care in, I don't know, 2010 or something. But is there ever a case, or do you guys have a mechanism where you lobby, influence, inform the insurance companies to say, this is a, that's one of the things that we can do, where we say, this is an efficient pathway, MRI first, and it brings these benefits. Art looks like he might have a heart attack. Just throwing it out there from the
in the United
future.
States, the first step of that is called a peer-to-peer. And who loves peer-to-peers here? Raise your hand. We have a mechanism.
I just wanted to add one more thing. We'll talk about this during the course of this meeting, but I know a lot of people in the room participate in this. We wrote a nice real-world data summary on HIFU and cryo. We had a lot of data, and some people ask, well, why does the Focal Therapy Society do this? Well, one of the reasons, 2 reasons. Number one is we want to capture what we've done in the past historically, even though we've moved towards focal. But second, at our institution, one of the insurance carriers decided that cryo now would be considered investigational. So you have something that's been FDA cleared, you have something with an actual CPT billing code that now goes on the investigational side, and you ask the question, why? And the response is, well, because it hasn't had a head-to-head trial with radiation or surgery. And then, wait a minute, that was like 30 years ago, but I guess they have the liberty to do that. So how do you move this field forward? And I think, Herb, you mentioned that you had some issues in New York with some of these carriers too. So, how do we move this forward when we have all these different stakeholders? We have government, we have FDA, we have CMS, private insurance, patients demanding this, and then physicians who, at least in our country, have some degree of flexibility and latitude to determine what's best for the patient.
I want to pop in with just kind of complimentary thing. I mean, I really appreciated Dr. Lepore's comment. I think it really needs to be said and focused on that. The way I interpret it, I think anytime you do any study that takes 7 to 10 years to read out the primary outcome, you are going to have problems with that study, no matter how extremely well and thoughtfully and well-resourced it was developed 10 years ago, right? And so I think that for localized prostate cancer, which is the space that most of this work focuses on, outcomes of metastatic and certainly disease-specific survival is problematic, and I think that, and this will just be kind of a recurrent issue, and I think some of the ways that the FDA has gone around it is by using ablation, but like a non-cancer outcome really introduces a lot of challenges, and I think that the field of management of localized prostate cancer, it's not inherent in like the randomized clinical trial for prostate cancer, right? Like for metastatic prostate cancer with 1-2-year outcomes, randomized clinical trials as the like standard of care for making decisions makes a lot of sense, right? But for something with 10-year outcomes, like it just won't. It doesn't, and it won't. And so I think it's been great to see the FDA has had some flexibility with like the ablation outcomes, and then they qualify it with certain things, and I think that that's like a lot of progress. But I think both the specialists in the community, in collaboration with the government and all these people who make these decisions, if there could be a primary outcome that is, it's not going to be perfect, but is a 3 to 5 year outcome, I think that will move the field forward, right? And then you can do randomized clinical trials because studies will, just the logistics of it will work, right? Anyone that's designed studies that has like 9 to 10 year outcomes, you just know it's going to be a problem, even though it makes sense today. So I love, the question is like, what are the chances of coming up with a cancer outcome, not just like an ablation outcome, but like a cancer outcome that is an intermediate outcome? Like, what are the chances? And understanding that there will never be one that's like a good like it will never be the same as like cancer-specific survival, but understanding that that is just inherent in the space that we live in here. What would it take to kind of come to a cancer outcome that is accepted and can become the basis by which people design these future studies and move forward the field?
Yeah, so I don't know if you're here for the talk earlier this morning. We sort of laid out one potential design, which was also detailed in the 2020 paper regarding the 2018 workshop, which was a design in which a low-risk population could be randomized to active surveillance or active surveillance plus focal therapy with a primary endpoint of an objective criteria that would translate to everyone would agree that would translate to a whole gland or definitive therapy. So whether that's Gleason progression or other markers, clearly following patient-reported outcomes, sexual, GI, urinary function. And again, I don't think they would need to be powered necessarily to look at those later oncologic endpoints, but at least the ability to follow for long enough and have enough events that you could look at it descriptively and have a good sense that you're not adversely impacting later term oncologic endpoints.
So it's an interesting point that Dr. Siddiqui brings up, and as sort of a follow-up to that, all of the devices that have been used and discussed this morning are all considered to be Class 2. They are 510K cleared. They don't undergo any PMA. And for a Class 3 device, those require a much more rigorous process for safety and efficacy, and with that, a much higher level of data. And so, to Dr. Siddiqui's point, in order to sort of bridge that gap, is there a role, is there value from the FDA standpoint to go through that process from a Class 2 to a Class 3? And with respect to the type of data that's being generated, is it possible to undergo that type of PMA for a Class 3 device, a cancer-specific device, without having randomized trial data?
Sure. So there's a myriad of variables. We're speaking in extremely broad strokes, but the process, as it works, is that we don't lay out what a device should be indicated for or for how it should be regulated without understanding completely how it works. So a company will come forward, will evaluate their claim and the data supporting it, and then we'll make a consideration. But in terms of accepting data, well, first, depending on the indications that they're asking for, it could be further specified by the level of risk for, through the Gleason grading score or whatnot. That's the first part of it, and the second part is the data to support such an indication. We're open. We've, as I said, we've published guidance on real-world evidence. We're open to considerations that we've all discussed today. This is very much a conversation we're willing to have on a case-by-case basis as each sponsor brings forward their device design for such indications. So, we're listening. We're here to understand what the needs are, but it has to come forward to us, and as it's kind of stated, can it be Class 3 to Class 2? That's a very, very open-ended question. I mean, it depends on the nuances of the indication. It depends on the risk we equate the indication to. And Class 2 is a low to moderate risk, a risk classification, versus Class 3, which is high risk.
Thank you.
I guess I think we'd all agree that we treat prostate cancer to prevent metastases and to prevent prostate cancer mortality. And if I'm not mistaken, I didn't have my hearing aid, I didn't charge it. But I thought that I heard that there wasn't a case of metastases in a 10-year study that was done, I think, at the UK. So obviously, if that's going to be our endpoint, it ain't going to happen. The other thing is that in this disease, and I think the complexity, and I'd ask Dr. Moore for her thoughts, we are balancing cancer control and quality of life. That's what we do when we do a radical, how much radiation, adding ADT, focal therapy. And so how do you get an endpoint that balances those two? You know, I will tell you, as a resident, Dr. Scott, who was the chair before Walsh, he said, you know, when penicillin came around, you didn't need no randomized study. The damn stuff worked, right? We don't need a randomized study. So if you tell me our goal is to get every cancer cell, there's no way focal therapy is going to equal a radical. And I can tell you, there's no way that the functional outcomes of the quality of a radical is not greater than focal therapy. And the real issue, and I'd ask Dr. Moore, how do you balance those? So first of all, if you have an oncologic outcome, what's it going to be? You know, with focal therapy, it's based on biopsy. With radical, it's based upon biochemical recurrence. Are those equal? Is a PSA of 0.2 after a radical equal to a biopsy after a focal? Although I think that Mark doesn't feel you need to biopsy, you just need to do imaging. And the question is, so if you do one focal therapy, can you do another one? Can you do another one? And so I can already see the problems in that randomized study impacting treatment. And then, of course, we'll say, well, incontinence, that's because in the UK they don't know how to do this operation. If they came to the US, we don't have incontinence, right? And so I just see, I want to come back to the challenges that we have in this disease is there is no single endpoint. There's no endpoint that we would agree functional versus and oncologic within that domain, and how you balance those two. And I'll tell you, there are patients that'll say, doc, just get this damn prostate out of me, this cancer. And others will say, hey,
whoa.
You know, whoa. So the issue is a randomized study is going to be a challenge in helping, you know, bring this down to the patient level. And I think that's the challenge we have, is we have two competing endpoints, and how you value those is really a patient-driven decision in many ways.
So just on the same exact line, in the PROTECT study, I don't think they biopsied or the radiation candidates or in the PROTECT study, right? And those are positive out 2 years plus, and they use a biochemical endpoint, but they're really using the justification for treatment to decrease the incidence of metastatic disease and death from prostate cancer. So that's the challenge you're posing, is that they're not all this, the bar is never the same in any space.
And there was no criteria for who got a biopsy, right? I can tell you PSA is worthless in this follow-up. So in that study, in the PROTECT, who got a biopsy, who didn't? It wasn't legislated.
Dr. Moore.
So you raised some very good points. And there've been some really interesting discussions for the PART study. So we are randomizing people. And I guess we have taken the approach of what really makes a difference to a patient. So I think for a patient having radical treatment, if you need radiotherapy or systemic treatment, that's a big deal. So minor fluctuations in PSA that you're monitoring, we didn't count those in the sort of propensity analysis, and in part it's additional treatment. Now for focal, many people consider that a second go doesn't make much difference, especially if it's in the same place. So 2 focals are allowed. And of course, my radical prostatectomy colleagues went, that's ridiculous. We can't take out the prostate again. But actually, what's a transition to a different treatment? Because for a focal patient, a second focal, very little functional impact. But having a radical after focal absolutely impacts. That's where we've set the line.
And then you come to the U.S. and our radiation oncologists say, we need to do your radiation before it's 0.2 in order to save your life, whereas there in the UK, you say, hey, you know what? Whenever we get around to retreating you.
And I just want to add to Caroline's statement, because if you look at kidney, you take a different organ. If you compare partial nephrectomy to ablation, well, clearly it's, partial is a little bit better oncologically, but then if you say you're allowed to do a secondary ablation, then it's more equal. So, to me, it's a little bit unclear why in the kidney you can follow up with another ablation, yet in the prostate, no one really talks about or uses that as an outcome, because I think that's very reasonable and realistic if you get a second ablation and hit the same bar.
I have a pet theory on that, so I'm going to put it out there, which is that taking out a kidney is a complex operation, doing a partial is a more complex operation by the same people or a sub-cohort of the people who do radicals, and I don't do any kidney stuff. But with focal, for prostate cancer, it's a completely different skill set. So it's a much more disruptive process and concept than partial versus radical, I think.
Yeah, if I could just get back to Dr. Lepore's concerns about the utility of randomized trials. In a comparison of focal therapy and active surveillance, a randomized trial wouldn't be to pick an absolute winner, because as you described exactly correctly, it's a balance. It's a very difficult balance. But it's only by doing the randomized trial that you get the information that can allow people to make the right decision for them. For example, the 19% rate of transitioning to definitive therapy in Dr. Moore's retrospective HIFU analysis, how does that compare to the 36% at 10 years in Toronto, when I think only a third of those patients progressed due to grade progression, or the 25% progression at 5 years at Hopkins, when I think only two-thirds of those guys progressed to definitive therapy because of grade progression. You're only going to know if you do a fair, balanced, randomized prospective trial, but you're not going to ever have an absolute winner. And just because you can't get the survival data, clearly you need to rely on surrogates such as, mainly I would say grade group progression would be the most important one. You're not going to get to survival. I mean, you might get to metastasis. In 10 years, they did get to metastasis in PROTECT. There was a significant difference in favor of treatment versus monitoring, something like 3 versus 6%. So it's not that you'll never get there, but well before you get to 10 years, I think you could get important information that would help people make the decision to choose between two options.
Please. Hi, good morning, and thanks to the panel. I'm Jonathan Fainberg from Sloan Kettering Cancer Center. Just wanted to bring us back to sort of some oncologic definition of endpoints. There's been some sort of debate and discussion over what would be a meaningful endpoint that we would all agree defines a progression event in terms of these trials. And I was wondering if the panel had any sort of concrete thoughts. Grade group progression obviously has been suggested a number of times, but we survey a lot of patients now with Gleason 7. So I was wondering, is it millimeters of pattern 4 on a biopsy? Because when we're defining these trials, what we're having trouble with is picking an objective endpoint that's oncologic, that's going to be widely accepted as a definition of success or failure. Thank you.
So it's an excellent question, and I'll let the panel respond. And conveniently, Professor Moore is also at the microphone, because I know that her group at UCL has done a lot of work with PIFAB and using imaging as an endpoint to drive definition of oncologic outcome and treatment success or failure after ablation. So
I guess though specifically in this country, because I'm not sure radiographic is going to be accepted as a widely accepted definition of failure or success.
I mean, I think we're agnostic. I mean, I think it should be what the community believes should trigger a whole gland definitive therapy. So I mean, I think that has probably evolved over time. My sense is that most of the community agrees that grade group progression is the most clinically relevant. But if there are other markers that the community feels are appropriate to trigger objective criteria that can trigger a definitive therapy, I think we're open to it. I think it's what we're trying to prevent is, and what would make the trial difficult to interpret is going on to a whole gland definitive therapy in the absence of those prior objective criteria.
agree. So how do we get the community to agree on what this is? Because there's been like a lot of suggestions here, and this is a panel or a room of people who sort of all think like-mindedly about this kind of stuff, and I would argue we would have a hard time all agreeing on that.
I mean, I would just add that's why we're here. I mean, we're looking for white papers, guidelines.
Yeah.
We're looking for consensus from the community, as Dan said.
Very nice. Well, thank you guys again. Appreciate it.
So I was just going to comment on, you asked what the difference between lorry clots is 30% progress on surveillance compared to 20% after focal HIFU. And I would say that the difference is that they got into focal HIFU by being MRI defined, so probably overall a higher risk group of people than the people in lorry clots as surveillance. And I think actually that was one of the flaws in the TCAD study design. The men who had surveillance weren't allowed to have an MRI, so I didn't recruit at all. I thought it was not ethical in my setting to get people enrolled in a study with no MRI for surveillance, in London at that time. So, and in terms of however, I don't think MRI is the answer for where should we set our things. So we've put out a proposal for a system called PIFAB looking at focal imaging after focal, but I don't think we should be using any PIFAB or any MRI criteria to define failure. I think we should use MRI criteria to decide who needs a biopsy, and that a combination of biopsy and other things would define who needs more treatment.
Please.
Matthew Bernig from Profound. So apologies if I'm speaking out of turn, being from industry. Very great conversation. Thank you. I think there are lots of examples where you don't need randomized trials. Smoking, for example, there are no level 1 randomized trials, smokers versus non-smokers, and clearly there's overwhelming evidence to suggest smoking is bad for you. I just want to make a point that there's a lot of discussion to the FDA around this question of randomization and endpoints. From my perspective, the FDA has been very open to discuss various pathways forward, which they've done with the tool claim versus the treatment claim. I think one important part that Dr. Moore touched on is when she did the precision trial, that she said we already knew what the outcomes were going to be, but we had to run the level 1 trial to convince others and impact guidelines. So I think one key piece that's maybe missing from the panel, from the discussion, are who are the NCCN guideline panel members, who are the AUA guideline panel members, who are the EAU, because without the guidelines adopting some of these things, from our perspective, that's kind of where there's some difficulties. The FDA has actually been quite flexible in demonstrating a pathway forward. Obviously, the physicians in this room are supportive of focal therapy. There's a lot of publication now with patients in the thousands. Why is that data not sufficient to then get into the guidelines to say, yes, focal therapy or ablative therapies, are good treatment options for men with certain disease states? So I think that's, from our perspective, that's a huge piece that's missing, all the way through to also positive coverage. That's another key piece that I think Dr. Palashik touched on as well. And so at Profound, we are sponsoring a level 1 study randomizing to prostatectomy. A lot of it is geared towards guidelines and positive coverage, even though we feel like we know what the answers are going to be already in that trial. Thank you.
I can help a little bit with this. Great question, because it makes sense, right? We're not governed medical practice by the government, right? So we are governing ourselves. And having been on the guidelines committee for prostate cancer most recently, it just came out at the AUA this year. And a lot of other members of the guidelines committee are actually in this room. It is a multi-year process with a high level of, I would say, critique of the literature. And it's taken very seriously. Everybody on that committee volunteers their time to put forth a paper that guides so we take it very seriously. And so at every level of evidence, we use all the information possible. and then have criteria to determine how well that should match our guidelines. So it is being thoughtful, and it's not always randomized trials, as you said, but the folks on those guidelines spend many hours. I put in many hours in those guidelines to make sure that we guide our physicians the best we can. We really do.
Dr. Robertson.
Yes, I just wanted to say a few things in reference to the society and its role here today. We really appreciate the FDA coming and spending your time with us today, but I think we should also think about how we can help the FDA. A couple of issues. Practice management in this country is guided by society guidelines and also by insurance provider guidelines, which sometimes are cryptic. And in my discussion with medical directors of several major insurance providers, their influence on approval is really on two things. One is North American literature. My apologies to my esteemed colleague in the UK. And also patient request or patient demand. And that guides a lot of decision making. If we participate in the registry and we provide high quality data, that influences guideline development. But as we've heard, it does not require a prospective randomized trial. However, for regulatory approval, you need the best quality data possible. So defining the population, the ideal patients, the perfect endpoints should be our primary mission here as a society, if we can. Thank you.
Wow. If no other questions or comments, Dr. Rassenhead? No? All right, perfect. So we will go ahead and adjourn the Focal Therapy Society meeting. We'll pick back up at 1 o'clock, so we'll give you guys a little bit of time back to allow you to go get some lunch and then make it back to the room in time for the 1 p.m. session to start again. I'd like to thank Dr. Pinto, who has some closing remarks, as well as folks from the FDA for their participation, Dr. Parnes, Dr. Rassenhead as well. So thank you all so much.
I just want to give a special thanks to Dr. Rothberg, who's done a wonderful job handling this. He had to tackle this with family decision, things going on, work and other related. So Michael did a great job for this, and I want to acknowledge that. And then for Dr. Pulasic, who's running around heading this up, and for Art here, we have some announcement that we'll start. It's at 1 o'clock, about this afternoon, the evening.
It's 1 o'clock. The official program starts for our hands-on, basically working through cases, and you'll be able to see the different vendors and have experts and key opinion leaders help you understand the new technologies that are available for ablation. And we look forward to seeing you guys at 1 o'clock. Thank you. Hey, thanks. I mean, it was a pleasure. I know it's a lot to come down here to.
I appreciate you having us.
Oh, it's great. No, we really